Hello everyone. We are live on YouTube now. Thank you.
Yashodhan.
Yeah.
Good evening, everyone. I'm Dr. Smita Kare. I'm working as Manager Bioincubation at Venture Center, and I'm also leading BIRAC's Regional Bioinnovation Center, that's BRBC, and I'm the project leader for Center for Biopharma Analysis at Venture Center here. On behalf of Venture Center, BRBC, and CBA team, I welcome all of you who are there with us offline and those who are there with us online today. Thanks for joining. We are really excited to have an offline event after, say, 2019 now. So today we have gathered here to conduct the symposium, which is based on how to take biopharma products to the global markets. So to take them to global markets, we really need to understand the regulatory pathways. So we have speakers, eminent speakers from industry who will share with you what is the biopharma drug development pathway. Then they'll tell you about regulatory guidelines and then share their own experiences also. So now I welcome Dr. Narendra Chirmule, who would be chairing the session. So before I invite you, I would like to introduce you to our audience. Dr. Narendra Chirmule, he's a CSO acumen, he's a freelancer. He's a biotechnology professional with a music enthusiast. He has published and presented seminars on subjects of immunology, biology, vaccines, and contributed to development of vaccines and biologics. The drug development experiences include vaccines for cervical cancer, varicella zoster, rotavirus, and biologics for osteoporosis, rheumatoid arthritis, and platelet loss, breast cancer, and among many others. He has worked on basic concepts of gene therapy using viral vectors. He did his PhD on development of a leprosy vaccine from Cancer Research Institute, Mumbai, postdoctoral studies on pathogenesis of AIDS from Cornell University Medical College, North Shore Hospital, and taught immunology at the University of Pennsylvania. So I welcome you and over to you. It's okay, you can use
Yeah. So welcome everybody. Thank you, Smita. Thank you, Dr. Ramnath. I know he's busy in a meeting, but thank you to the Venture Center for organizing this event. When Smita and I spoke maybe a month ago, maybe a little bit longer, we felt like there's a lot of biotechnology and biological research that's being done in Pune. And we had started this in 2018-19 to start building this infrastructure in this room. And I remember coming to this room and having this symposium, and then, life changed for us. And maybe it was a good time to start. And it's always a good time to start on this kind of an occasion where we have this, now hybrid is going to be the norm, right? So welcome to everybody who is online, and please bear with us and please speak up or text up.
Yeah, in the chat box.
Yeah, if you have any problems or you're not able to participate in the conversation, please let us know. But I look forward to this exciting session. I am so excited to see the students who have come here in large numbers. Thank you for coming. It is, because of you, this place is going to become vibrant, right? You are going to take the future of biotechnology. So ask question, I said, right? Ask faltu questions, like ask questions, everybody. But let's have fun, right? Masti karne aaye hain. Khana milne wala hai na? Online wala hum log khana milne wala. So with that, I'll give it back to Smita to take off. And we'll have a panel discussion where we'll have more discussion.
Yes. And for our online audience, please keep posting your questions in the chat box for all the speakers. So I'll now start with a quick introduction of Venture Center to all of you. Yashodhan, can you see the screen full screen?
Yes.
Okay, yeah. So we are Venture Center. We are India's leading inventive business incubator. We are hosted by CSIR NCL. We are a Section 8 company, non-for-profit. Being non-for-profit, we are eligible for CSR support. We are now 15 years old. So we have 15 years of experience. We are the first incubator under CSIR, and we are recognized by DSIR and DIPP and approved by NST, EDB, and BIRAC. Those are DST and DBT bodies. So these are few recognitions which you see, which we have won on national and international platform.
Hello, Smita ma'am. We cannot see your presentation. We can just see your Windows screen.
Okay, let me share.
The list of the presentations.
Now, can you see the mission slide?
Yes.
Okay. So the mission of Venture Center is to nucleate and nurture. So by nucleate, we mean if you're a first generation entrepreneur and you don't know how to start your entrepreneurial journey. So at that moment, as well as nurture means if you already have a startup and at a certain point you realize that you need to raise more funding or you're looking for help for, say, scaling up, that time also you can come to us. And how do we help our startups? Is by leveraging the scientific and technical engineering competencies in the region. So we all know we are in Pune, which is Oxford of East. So we are well connected with our mentor community here who help our startups and us. So the focus areas here are healthcare, medtech, energy, environment, engineering, agri-tech. You can see that we support all inventive enterprises. So if you have a very good invention which can be protected by IP, then we support you. We also support spin-off from R&D institutions, and we are sector agnostic. So some unique aspects of Venture Center, as I told you, we are 15 years old now. We have supported 600 plus startups till date. 150 plus have worked on our campus as resident incubatees. We have supported 10 spin-offs. As I said, we have a highly engaged community of 200 plus mentors here, and we keep conducting events throughout the year. And these kind of events actually are a good platform to connect and network and learn from each other. So we provide high-touch mentoring support to all science and technology startups who are at early stage. By early stage, we mean either idea stage, proof of concept, or prototype stage. So 85% startups supported by us are still active. 90% of them are in the early stages. 75% plus are inventive startups, and there are then you can see more than 100 unique families of patents which have been filed. So very proud to tell you that 30% and more are women founders, 85% more are founders with very good work experience or academic background from premium institutes from India and abroad. In total, we have 50,000 square feet of incubation space, and we have created 9 unique resource centers here in Venture Center, where we are offering advanced services to startups. So you can see that almost 500 CR risk capital has been mobilized in last 6 years by our startups, and approximately 1,000 CR revenue has been generated in last 5 years. So Venture Center is here to provide comprehensive support to promote early stage ideas, startups, and entrepreneurs through its diverse and impact ecosystem. So this is a unique, complete ecosystem which we have at Venture Center. So we have dedicated lab spaces, office spaces, which we offer to entrepreneurs. Other than that, we do a lot of hand-holding by mentoring these startups, mentoring how to raise their funding. We host a lot of fellowship programs, and we are implementation partners for many of the funding programs by government and CSR. We have a very good library here. Then we have all advanced analytical facilities, and there are shared lab facilities. And I'll be happy to share more information, and you can always connect with my team members also to get more information on the equipment which we have. We also have support for technology commercialization. So we have an intellectual property facilitation center, which helps you to, file your IP. We have a TechX office, that is technology transfer office, so which does the matchmaking of technology seekers and technology providers. So these are some of the programs which we run at Venture Center. So these are the success stories which we are really proud to showcase. These are the first in India or first in world technologies by Venture Center startups. These are the spin-out success stories supported by us from CSIR, NCL, and ICER. But we are open to all academic organizations and they can approach us for our lab to market program. This is a quick incubating snapshot. So you can see that these are the different domains which are coming up at Venture Center: biomed devices, implants, diagnostics, environment sustainability, biopharma, agriculture, bio-IT, engineering, industrial biotech. And you can see that most of them are working in the healthcare domain, in the devices and implants area. This is the board of directors of Venture Center. Dr. Premnath will be joining us soon. These are our supporting partners from government, our host institute NCL, NSTEDB from DST, BIRAC, DBT, MSME. And these are our CSR partners. And we are happy that Dr. Kore is here from Intox. Welcome, sir. These are the glimpses of the facilities at Venture Center. So just to give you one example, if any startup who is working on a medtech product, then they can approach us and we can help them right from idea to taking that idea to POC stage. Whatever experiments they need to do, those can be done in our labs. They can make their prototype in our tinkering lab, which is well equipped. They can do the trial manufacturing in the clean room, which we have. The sterilization and packaging can also be done there. And to understand the regulatory pathway, we have a regulatory information facilitation center. So that team can help them understand the regulatory pathway and prepare for it. So that's all from my side about Venture Center. We'll be happy to connect offline with you during the networking session for more information on the facilities and services provided. So shall we begin with the session? So our first speaker for today is Dr. Taruna Arora, and she's currently at Coherus, US, but she has shared her pre-recorded presentation with us. I'll quickly introduce her and then I'll request Yashodhan to kindly play her video. Yashodhan, if you want, you can start sharing your screen while I'm introducing her. So Dr. Arora is Vice President, Biotherapeutics Discovery and Translational Research at Coherus. She is a R&D leader with a passion for immunology, drug hunting, and drug development to enable effective solutions for treatment of diseases. In 20 plus years of working in large and small biopharma, biotech, academic institutes, and VC firms, she has led initiatives and team efforts to deliver in multiple biologics, IND candidates, inventions, and approved drugs. Her interests lie in disruptive thinking, accelerated advancement of therapeutics to clinic for patients' benefit, and shaping corporate strategies. She is an active contributor to EU, US, Australia filings, BLA, MMA approvals, F2F, FDA meetings. So we are very thankful to her. She has kindly shared her presentation and she will join us during the panel discussion. Yashodhan, please go ahead.
Good morning, everyone. Thank you for the invitation to be part of the panel discussions after these overview talks. My presentation will focus on the process involved in drug discovery to IND filing, focusing primarily from the FDA perspective and also the industry standards that are used in the drug discovery process. Before I start, I wanted to provide briefly my background. I did my undergrad and master's in Delhi and then came to do PhD in immunology at Mayo Clinic, where I was studying interferon alpha therapy resistance mechanisms in multiple myeloma. Following my PhD, I went to Dr. K. Shuai's lab in UCLA for postdoc, where I was studying interferon and cytokines, other cytokine signaling pathways. Since then, I have been in the industry, both large, small, mid-sized pharma companies, including Amgen, and I have been part of from drug discovery to product approval as well as life cycle management. Currently, I am a vice president at Coherus Biosciences, where I oversee antibody discovery and translational research. So before you embark on the journey of biotherapeutic drug development, you have to arm yourself with the knowledge of target biology and disease area of interest. You need to ask why you're selecting this target. Is there something about the expression of the target in disease versus normal setting that make it interesting and indicate its role in the pathophysiology? What is the structure of the target like? Is it a soluble protein? Is it multi-transmembrane protein? Does it have cofactors associated with it? Is it GPCR or ion channel? Because all of this will determine whether your path to discovery and screening for drug candidates is going to be straightforward or is it going to have some challenges. Also, human genetics knowledge for the protein can be very valuable, particularly if there are mutations or deletions that may be known to exist for this particular target. One such example is PCSK9. Mutations in PCSK9 result in the hypercholesterolemia and high levels of triglyceride, resulting in these patients having higher risk of developing heart disease. This idea and these observations were used in the development of antibody drugs against PCSK9 to degrade the protein and to lower the levels of cholesterol. Repatha is one such drug which has been developed and is marketed by Amgen for lowering the cholesterol levels in high risk heart disease patients. Also, animal models can be useful for understanding the contribution of the target in a particular pathway or disease biology. Knock-ins, knockouts, or transgenic models have been used for these purposes. However, sometimes you have to be careful in interpreting the data because not all pathways are conserved from mouse to human. The second question you should be asking is, so what is the unmet need that you are trying to address with the biotherapeutics drug development? Is the drug going to have a better efficacy profile compared to the existing options, or is it going to provide a more safe option? Are you looking for cost-effective solutions by developing this drug? Which is going to be true for biosimilars because biosimilars have abbreviated clinical path for development, and they're also marketed at discounted price, thus providing more access to patients. However, for innovative drugs, the path for development will be longer. Therefore, it is very important to understand the need that you're trying to address by investing in the biotherapeutics drug development. The next question is what are the specific indications and what will be the path to first in human trials? Will you envision going into a refractory patient population for the clinical trial? The refractory patient population is the one which may have failed other approved treatments, and therefore this may provide another option for such patients. Or is it that there is no biotherapeutic against this particular target, and therefore your biotherapeutic can be first in class? Such example would be Aimovig, which is an antibody to CGRP receptor, and before that there were only small molecule or Botox-like treatments for acute and chronic migraine. So Aimovig has really proven to be a very efficacious drug with less frequent dosing for the treatment of acute and chronic migraine. If the desire is to have accelerated path to approval, then one can also consider orphan indications. In US, orphan indications are less than a patient population with less than 200,000. For anything outside oncology, your first in human trials involve healthy volunteers where you are studying trying to identify a safe dose and perhaps also some pharmacodynamics markers which would hint on the activity of your drug. Now, there are several modalities that one can use for the purposes of developing a biotherapeutics. These are listed here, and it really depends on the need or the underlying disease that you're trying to treat. For example, peptides such as GLP and insulin are being used as therapeutics. They are daily injections. These are the therapeutics for diabetes. Then there are therapeutic proteins, which are recombinant versions of the endogenous sequence produced either in mammalian or bacterial systems, such as EPO and Nipogen, which is GCSF. And again, if the desire is to have less frequent dosing for such proteins, then fusion proteins were generated that were either pegylated versions or high glycosylated versions of these recombinant proteins. So one example is Neulasta, which is pegylated version of GCSF, and then Enbrel, which is a FC fusion of TNF receptor used to neutralize TNF. Other examples are antibodies, which are large molecules greater than 150 kilodalton, and then there are other engineered versions of the antibodies that result in bispecifics or multispecifics, drug conjugates, and even small molecule conjugated antibodies like PROTACs. Besides these protein-based therapeutics, then we also now have cell therapy and gene therapy options for modality. And again, it depends on what is the underlying disease that you're trying to treat. So as the part of your drug discovery process, you should start first with a therapeutic product profile, which will include your design for the therapeutic modality, rationale for this therapeutic, criteria that you will use to identify your lead candidate, and the dosing regimen, whether it is going to be a sub-Q or IV administration or even less frequent dosing that is envisioned. At the same time, you should also consider what differentiates your biotherapeutic drug from those that already have been approved or where others might be working on against the same target. Competition is also important information to have because it informs you whether you can catch up or you can have a competitive edge over others. So once the target therapeutic product profile has been defined, you can start the drug discovery process. And drug discovery process really has 4 phases, starting with the lead discovery, when the candidates are generated and these candidates are then screened against a specific criteria you had set up in your TPP, and you are characterizing these candidates in vitro assays. From lead discovery, you go to the next phase of lead optimization. This is where you would have narrowed down the candidates, and a few candidates would be now characterized for potency, pharmacology, maybe also in vivo models. They will be evaluated for PK properties as well as for developability. And this lead optimization phase results in narrowing down candidates to maybe top 2 or top 3. And these 2 or 3 candidates can then be advanced to stable cell line development process with the intent to have a manufacturing cell line that will provide clinical material. So IND enabling studies are mostly done under GLP or GMP, and these include tox studies where applicable and also formulation stability studies. And at this point, you can determine whether or not you need to have a pre-IND meeting with FDA. Sometimes if companies that have already experience in going to IND and have had interactions with FDA before, they kind of know what is exactly the process and whether they can skip pre-IND meetings or there are any observation during their tox studies or formulation studies that may require a dialogue with FDA. So it really depends on your need. So for IND filing, you would need a data package that demonstrates that the product characterization that you've done as part of pharmacology is adequate, and that the product quality you're generating from your manufacturing cell line is consistent and can be controlled, that you have identified doses to be used in first-in-human trials. So it's going to depend whether you are doing a normal healthy volunteer study or you're going to go in patient population. Based on that, agency can have very specific considerations on what dose they would allow to be used as the starting dose. So let's talk about the drug discovery process briefly. For antibodies really, as I mentioned, they are highly specific and safe. Antibodies mediate their effects both through the variable regions, which are shown by these two arms, and also through the Fc region. So because of their structure, they tend to have better PK properties, as well as they can have better coverage for in the case of high affinity antibodies. They mediate agonism or antagonism, and they can also mediate Fc mediated effector functions such as ADCC, CDC, and they can also deliver drugs, small molecule drugs or toxins, to mediate cytotoxic effects. In general, the antibodies are well behaved in their manufacturing characteristics, particularly if we're talking about IgG class. Now, there are companies in US that are also working on IgM type of therapeutics, which tend to be pentameric in nature, and these can certainly require manufacturing considerations, which usually are not really, have been an issue for IgG class. Now, most antibody therapeutics that have been approved or are in development are humanized or fully human. When the first antibody was approved for therapeutic purposes, which was OKT3 against CD3 of T cells, it was fully mouse antibody, and it had a lot of immunogenicity issues. Therefore, could not be dosed after certain. It had a dose limitation because of its adverse events. Then came chimeric antibodies, where immunogenicity was reduced but still high because it still was 50% mouse or in its structure. So that led to evolution of humanized antibodies and fully human antibodies. Humanized antibodies maintain antigenic determinant regions from mouse origin onto fully human germline frameworks and fully human Fc. In terms of the human antibodies, so I'll skip this. In terms of fully human antibodies, there are display technologies such as phage display and yeast display that have been used for screening, for panning, and then identifying the candidates with favorable properties. Usually these libraries are either Fab or SCFV based fragments, and they require final formatting into fully human antibodies. Human IG transgenic mice and rat platforms are also used for generating fully human antibodies. In these mice or rats, the rodent immunoglobulin genes have been knocked out and they are replaced by the fully human variable regions. Thus, The antibodies that are generated in these rodent species will carry fully human variable region, and that can be then engrafted onto human FC to make them as fully human antibodies. But now the field has evolved even further. Now there are AI aided designs being developed for antibody therapeutics generation. And the reason the field has moved in this direction is because now you do have structural information for antibodies, you have structural information for the targets. So using the modeling, now the computational scientists can identify the CDRs regions that need to interact with the target depending on what you intend to do, whether it is just a binding that is required or if you're trying to block receptor ligand interaction. But computational scientists can model that, and then through a deep learning they can mature their models, and it can also be used even for in silico optimization for viscosity or solubility. So these optimized antibodies can then be, expressed and further characterized. This process is supposed to take shorter time for generation of candidates compared to display approaches or the conventional mouse immunizations or transgenic mice platforms. But that remains to be seen. Once you have screened for drug candidates, depending on your end goal for therapeutic in mind, you can also engineer them further to optimize the therapeutic profile. For example, if the goal is to have better specificity and safety, such as for blood-brain barrier, then you would engineer the antibodies for tissue-specific targeting. If the goal is to have better efficacy, then companies are working on bispecifics or multispecific designs to target more than one protein. Oligoclonal or polyclonal antibodies against COVID have been developed and approved by FDA to provide better clearance of COVID virus. If the goal is to have improved cell killing or cell depletion of certain population, then you can consider drug conjugates or effector cell recruitment, such as T cell recruitment, or maybe even enhanced ADCC modalities. If the goal is to provide fewer doses for administration for patient convenience, then the companies are working on co-formulation or developing higher concentration of the products than has been achieved so far. So the field is really moving and advancing very fast, and I think we are going to see some of the evolution, particularly in the field of multispecifics and also co-formulation. So as discussed before, you can engineer your antibodies for the desired therapeutic profile. While taking all the considerations for efficacy, safety, or combinations, co-formulation are all highly desirable attributes we want to see in the drug candidates, you also have to take into account PK and manufacturability attributes. So antibodies, just like other proteins, they can undergo modifications leading to oxidation, deamidation issues, or maybe even aggregates. So that can result in product heterogeneity, that can also result in an impact on stability and product quality. So this can eventually result in regulatory concern, particularly when it comes to product specifications and safety concerns, and also it can impact your cost of goods, especially if you may have to get rid of product heterogeneity such as truncated molecule or aggregates to generate highly pure product for clinical use. In addition to assessment of developability for manufacturing purposes, one can also incorporate immunogenicity risk assessment during the drug discovery process. And in this process, you can use in silico tools as well as in vitro tools, and if at all there is additional need, you can also utilize in vivo tools. However, most industries are focused on in silico and in vitro assessment for immunogenicity of protein-based drugs and antibody candidates rank ordering. So again, this is really dependent upon your purpose. If you have multiple candidates for antibodies to choose from, then you can use the in silico tools to rank order candidates. If you're developing a modified version of an endogenous protein, FDA will have concerns about the immunogenicity, so you can perform an in silico assessment to understand if there are any new T cell epitopes, and also then to come up with a plan for managing the risk. So in general, the risk assessment would really consider whether there is a high risk that we talked about, for example, engineered ligands, or it is low risk therapeutic such as antibody. So once you are through the discovery and optimization phase and you have identified your top 1 to 3 candidates, then initiates a stable cell line development and the studies for formulation as well as any GLP tox studies that may be required using the stable cell line material. This is the time you have to make a decision whether or not you need a pre-IND meeting or not. So pre-IND meetings are not necessary, but they can help de-risk your IND submission. You should refer to regulatory guidance for countries where you want to file IND. You may want to use pre-IND meeting to get alignment on your pharmacology package as well as on your clinical trial plan for the dose, for the indication, patient sample size, and patient diversity. Also, you may want to seek feedback on the CMC activities that you have planned and the analytical assessment that you are considering, just to make sure that it's not like agency may be anticipating more than what you have already planned. Also, you can get an alignment on the tox study of species and even determine whether you can skip the tox studies to start with a MABEL dose in clinic, and what would that MABEL dose look like. The successful IND really depends and hinges on your readiness with clinical material. That is, you have been able to manufacture a consistent, stable, high quality drug product that is safe for administration in patients, in humans. That's really the most important thing in regulatory agencies' mind, especially here with FDA. So key steps towards clinical material readiness would be to start stable cell line development, leading to research cell bank, and this is the time you would also perform cell line stability on clonal cell lines and prepare master cell bank, resulting in drug substance and eventually in drug product. And I will let my other colleagues discuss this more in detail, but this is just general big picture for the cell line development process. And finally, I just wanted to show this slide to show that innovative drug product process is, it does not just stop with the IND submission and approval. After IND and initiation of your phase 1, there will be other larger clinical trials that would need to be conducted, and then the whole package of clinical studies and manufacturing is reviewed by FDA, leading to product approval. So with that, I would like to thank everyone and look forward to the panel discussion.
Hello?
Sorry for those technical glitches. I would like to now invite our next speaker, Dr. Jyoti Iyer. Dr. Iyer was with Biocon, and now she's working as a consultant and researcher. She has a technical expertise of all aspects of CMC in developing therapeutic biosimilar molecules from early stage until regulatory approvals. She has worked on purification process development, scale down and process characterization, scale up, technology transfer, process validation, viral validation and troubleshooting, defining the CQAs, QTPP, and steering process development around this knowledge, analytical and characterization tools and technologies. She enjoys work around challenges by creating and influencing creation of systems and processes within and outside a span of control. She has a talent to build a connect at a personal and professional level, thus managing complex challenges through active participation, and can engage closely with stakeholders and senior management, and is passionate about teamwork and mentoring people. So with this short introduction, I welcome you for your session. Would you like to use the collar mic? Set it up. Yashodhan, can you see the presentation now?
Yes, can you just go on to the next slide?
Yeah. Is it
Okay.
moving?
Yes, it works. Thank you.
Okay. Afternoon, everyone. Thank you, Smita. Thank you. Yeah. If you want, you can use the color. I don't think So thank you, Smita. Thank you, Venture Center, for organizing this, and thank you, Naren, for inviting me for this. So very quickly, I'll just speak about what I have in the slides. What I've tried to do is give a very brief overview about the CMC, or the chemistry, manufacturing, and controls in the CTD. That's very brief. But what I'm trying to do here is talk about the learnings that I personally had from trastuzumab, the approval of trastuzumab, when we were interacting with the regulatory, when we were preparing the dossier. So what were the key points that we learned through experience? And I've also tried to include a few slides on the trastuzumab biosimilar. This is actually present in the FDA website. So what I have here is just a few representative slides, but if people want to go through in detail, this is available on the FDA website. So again, just a brief introduction about what are biologics. We all know that biologics are not chemically synthesized, and they are manufactured in living cells, making them so complex as we know them, right? They are not like aspirin or any of the other small molecules. They are quite complex in structure, whether it's insulin or monoclonal antibody. Again, the same, but just to specify that the heterogeneity is mainly coming from the post-translational modifications. So they are not homogeneous in comparison to the small molecules, which are fairly homogeneous. But why are we really working on biologics or biosimilars? Is because my personal belief is they have revolutionized the treatment of chronic diseases. So people are having an opportunity or an option to go for safer treatments, which will have fewer side effects. And also, definitely the market is requiring more of these drugs now. So coming to the CMC, as in a CTD document, that is the common technical document, this is a format which is harmonized across the different ICH regions like Japan, Europe, FDA. All of them follow very similar format of the document. You have different modules. The module 1 is general information. Module 2 are summary. The module 3 is a CMC section, and module 4 and 5 are non-clinical and clinical. So in the module 3, there are two different parts to it. First is a drug substance, and the other one is a drug product. And all the details of the CMC are to be incorporated in a CTD document in this particular section. So you start with the structure and general properties. And here comes the crux of the manufacturing information, starting from the process development, which is included in the manufacturing process development section. So there are defined sections that you have. So all the process development and other knowledge that you generate during R&D is included in the manufacturing process development section. And you have information about the manufacturing process and process controls. I'll try to specify a little on the process controls. So when you work on a manufacturing process and you start the batches, you will realize that there are certain key parameters that are critical to maintain the quality of the product. And if you don't maintain or if you don't control those particular process parameters, then the quality of the product can go off. Like, for example, pH of certain buffers or conductivity and so on. So what are those parameters that are critical for your process? You have to identify them. This is generally done during the process characterization studies. And then you have to set limits and specifications around it, which you need to include in this particular section. Then you have also control of materials. So control of materials are basically how do you control the inputs, the raw materials that go into the manufacturing process. What is the strategy? What is the approach? How do you control this? That information goes into the section. And also the intermediates, process intermediates, all of those control process all goes into the manufacturing section. The next key section is the characterization section, wherein you have to include all possible knowledge about your molecule, that is structural,
physicochemical, biological, about the impurities. So this is a very important section because this is the one that helps you determine or helps the agency determine what kind of impurities you have. Are they related substances or are they substances that can be potentially immunogenic? So the more you elaborate on this particular section, if you have some differences, but if you know what those differences are, and if you're able to identify and explain to the, in this particular section that they are not going to be potentially immunogenic, you need to generate data around it, but this is also a very key section which will help you even if there are differences. Then this is the control of the drug substance, which is basically all the QC related, all the quality control related information. So here comes the analytical methods. How did you develop them? Very minimal information on development, but a lot of information is required on the validation of the methods. So the qualification and validation of the methods. How did you do your validation? What are the parameters you used? The data from the validation, everything goes into this section. So there's a lot of importance that is also again given to this section because it determines the overall quality and how you're going to maintain it consistently. Because this section will give them the confidence that you have the tools to consistently maintain the quality that you're producing. So the strength of this validation package that you provide, the method validation package, will help the agency understand whether you have strong tools to determine batch after batch how good your product is going to be. Then the most important, or again, the key is the reference standard. What is the reference standard with which you are measuring your batch against? So there is there's a complete there's a paper that talks about how to determine the reference standard, how do you maintain the reference standard. There's also a lot of FDA articles on how do you work around the reference standards. Then you have the stability section. Again, another key section. Here you need to provide the data from the IND batches, all your development data also, some summary about the development data. You can use pilot scale data. You also need to include how you're going to do the post-approval. So once you get an approval, how would you doing your stability? What kind of stability would you be conducting? What tests you would be doing and so on. So there is a lot of back and forth here with you and the agency. Once you submit the protocol, they may agree to it or they may say, no, this is not enough and we need more. So there is definitely a lot of discussion from the agency once you submit these protocols. So this is a stability commitment. So whatever sections or information is provided for the drug substance, we have to do the same thing again for the drug product also. So all of this, manufacturing of the drug product, characterization of the drug product, control, how are you controlling the drug product, stability of the drug product. In addition, you have the container closure system, which is very unique to the drug product. So even if you have the drug substance going into the drug product and you are not doing any particular modifications to it, that you're not lyophilizing, it's just the drug substance that goes into the drug product. Even then, you're expected to have characterization data and all the other data, even for the drug product section. It's an extensive section.
Yes, wait for that.
So again, as we saw in the previous slide also, a summary of how the drug developed here. What I've done is I've also included the ideation, which happens for the novel molecules. But otherwise, then you have the cell line development, synthesis, gene synthesis, followed by cloning, and then cell bank. You have the upstream process, downstream, that is the protein purification, formulation, DP, and then the actual DP vials. So where does this complexity of a biologic actually come from? It comes from the fact that there are large number of amino acids. I mean, typically you have for a monoclonal antibody around 1,300 amino acids, and all these modifications, right? You have deamidation, oxidation, fragmentation, pyroglutamate formation. A lot of these modifications, some of them can cause, the loss of efficacy or create safety issues and so on. So these modifications are the reason for the heterogeneity that is observed in a biologic molecule. And that is also the reason why you have to characterize the molecule so very well to understand the heterogeneity that you have. If it's a biosimilar, then you have to show that the heterogeneity that is present in your molecule is similar to the heterogeneity that is present in the innovator molecule. So from here on, what I have done is I have just summarized a few points, around 10 points as key learnings and what I think are good strategies to have when we are developing the CMC for a biologic. Since that was the topic, strategies for CMC. So I would put the first one as identify the critical quality attributes, irrespective of whether it's a novel molecule or a biosimilar. I think that it's good to identify what are the critical quality attributes that we need to start working on. Is there any modification that can potentially impact the safety or immunogenicity? Then that kind of pushes back the development significantly. And hence, it's important to know whether any of these modifications can have an effect on the safety or immunogenicity. Potency is still manageable because you can purify and remove it and so on. But to prove that any modification that can cause an immunogenicity is completely cleared out and there are no traces of it remaining becomes a very challenging process. So the first one would be identify the critical quality attributes. Importantly, figure out whether they have an impact on any of these and start working around the QTPP. So what is the target product profile? And this will keep evolving as you develop. I mean, it's not necessary that you need to have a well-defined QTPP in the beginning, but to start with a working QTPP is good to have. The other important one is analytical methods. I don't know if any of you are from QC here, but I am speaking on behalf of all the process people, process scientists, because this is something that we struggle with, that if we don't have robust analytical methods to start with, then we end up figuring out whether the variability that is observed in development, is it coming from the method? Is it coming from the molecule? Is it coming from the process? We don't know what's happening if the method brings in a lot of variability. So to have meaningful data, that is, make sure that the data is giving you meaningful information from the development, during the development, if you need meaningful information, it's good to have robust analytical methods right in the beginning. And also one of the challenges, if you don't have these methods, which are good or robust, then the development data cannot be used. I mean, you end up not using the development data during setting specifications because the variability is too high, and then you have changed the method or tweaked the method as you progress. And then that data will not be useful for setting the specifications. So preferably try to get to a near close analytical method right in the beginning. I've just given some examples. We are aware that the cell-based assays are quite variable, but the HPLC and other purity methods can be quite, I mean, the variability can be minimal. So that, like, for example, if it's an SEC HPLC or a CASDS, try to maintain it between 10 to 15%. Don't start with like a 25 to 30% variability there. So this is an example. I think this is from our paper, and so you can see here that the innovator is much more variable as compared to the biosimilar. So we don't know whether that is because of the region and the difference, or is it because of the method itself. For a biosimilar development, another critical point is please source and identify the reference product lots. So to set the development targets. So this is also from our own experience where we set very tight targets right in the beginning. I mean, we would have, if we had limited lots, then what happens? You set tighter ranges for your development and it becomes a challenge to meet those targets. Whereas if you source more lots, if you are able to, because the variability depends on where you're sourcing it from, what is the location of manufacturing, the scale of manufacturing, what could be the age of the batch when they were filling the vials, or a lot of other the logistic variables also, like when are you sourcing it? Like in India, for us, we don't necessarily get the early vials, right? We would get it near expiry and so on. So all of these variables would add to the variability of the reference product that you're testing, in addition to the method variability. So you have variability from the innovator itself, and then you have the method variability. So the good thing to do is, if you can buy more of those lots, source as many as possible, try to look for different expiry dates. One thing we also realized is it's good to incorporate quality right during the clone development. So when you are setting up your clone development, biosimilar clone development, it is good to define or select the lead clone based on the quality parameters. That is, not a lot of them, but at least the purity, like purity, glycosylation. Are there any unwanted glycosylations? Are there any glycosylations which could be challenging to handle, like more sialic acid, more mannose, less mannose, and so on. And it's also very important to establish the monoclonality during the cell line development. So whenever, if you're doing it in-house, well and good, or if you're talking to a CDMO who's doing this for you, ensure that they have the facility to do the proof of monoclonality, which is basically what I've shown here in this picture is one cell dividing into two and then two into so on. So this is very critical, especially now because it's an FDA requirement. And then this is something that I discussed in the beginning also, well-defined and controlled manufacturing processes. Here, since you have so many testings, right? I mean, almost at every step of the process, you would have a testing, whether it is cell culture, purification, formulation. Every single step, you have some or the other in-process testings, right? Some of the, I would say, things that people do is we try to put tight limits, or we have too many tests. So what we realize that it's better to have realistic number of tests. Don't overdo it, but not minimal also, but try to find a balance, which is what is necessary to ensure the product quality. So set realistic tests and limits that can be managed during the commercial process, because then the opportunity to change them during the commercial process becomes quite difficult. So the other thing is we have to avoid too many deviations. So the tighter the limits, then the deviations can be more. Again, the detailed process characterization studies are very critical for this. As I mentioned, comprehensive characterization and testing is one of the most critical requirements, especially for a biosimilar. For a biosimilar molecule, this kind of becomes the backbone of your package. The more details and information you provide about the molecule, along with the reference standards or the innovator molecule, the stronger your package becomes. So extensive physicochemical and biological characterization, and multiple tools, that is orthogonal tests to determine the same parameter. It is anyway mandatory to use statistical tools if you're doing a biosimilarity assessment. Both FC and FAB, even if there is no FC function, it is important to show that there is no FC function. So even the function not being there, it is important to show that the molecule that you're working on does not have an FC function. And it is also important to show, as I mentioned, that there are no differences between the drug substance and the drug product. That is, when you are making the drug product, suppose you have a processing, let's say lyophilization. It is critical to show that the lyophilization itself does not cause deamidation or aggregation or any change during the drug product processing. One thing that people can miss is the extinction coefficient. So since this is important for measurement of the potency, because it's directly linked to the concentration, it is a good idea to do the extinction coefficient upfront to find out whether the extinction coefficient of your molecule is similar to the innovator or not. Yeah, so this is kind of my favorite topic about documentation. I actually have 3 slides only on this topic. So because we, for some reason, don't believe that it's important, as important as doing an experiment, we don't want to believe that documenting it is as critical, right? So we have learned in a very hard way that contemporaneous documentation and GLP practices, even in R&D, even when you're just developing the method, doesn't matter if you're developing the method the month 1, doesn't matter with like the stage 0 of the project, it is important to have some kind of documentation, some contemporaneous documentation. It doesn't have to be too stringent, but make sure that you are having a contemporaneous document. And a good consistent quality system is absolutely must. So all the development data, process characterization, analytical development, everything has to be as per the compliance. So there is a lot of information available about this, which is, it's summarized as ALCOA. Basically, it has to be attributable, legible, contemporaneous, original, and accurate. So the raw or source data, for example, original means you should be able to source a raw or the source data. That is, you have run an HPLC, you've written down a notebook, but you don't have the electronic copy or even a hard copy of the HPLC data, then the data is not valid. So the original data from which you're transcribing it into a notebook is as important as your notebook itself, because they look for that data. It could be anything simple also, like for example, weight. As simple as that, it is good to have a logbook recording of that. Yeah, like as I mentioned, this is something that we ignore so much. So we don't even realize. So this number that is written here, we don't know whether it is 24 or 29 or what is exactly the number is, right? And we really, as I said, we really learned it the hard way that some of these things can cause a lot of pain. It's just that it becomes more and more challenging and difficult. Again, a few more examples. Right? Legibility is so important. Making sure that you're not, even if you strike it or cut it, it's so important to make sure that you're cross-signing it. So if you're cutting it today, and after 2 years, somebody else is looking at your notebook, but you're no longer in the organization, then that data it becomes very difficult to come around and correct the data again. So these are practices that needs to be incorporated upfront. So even if it is startups, my suggestion is that because the data is going to be used tomorrow, incorporate documentation practices and systems as much as possible. Avoid pencil, marker. If a marker is available, we write with the marker in the book, right? So these are things at least we realize is not a good thing to have. And audits are not only QA, audits are both technical and compliance related, especially for approvals. Later, when the GMP audit for the commercial processes start, it is more QA related. But the first time the product audit is a mix of technical and compliance. So a sound technical knowledge or discussion with the agency is very critical. That actually helps them understand what is the science behind the development. So there's a lot of team effort here. I mean, all the teams have to actually work together and make it happen. So, I mean, again, this R&D sometimes believes that our part is done. We have handed it over to manufacturing, and we don't have a role to play. But it actually, till the end of the approval, R&D has also a very critical role to play here. Regulatory processes are very, very challenging, but actually the regulated markets make it a little more easier because they allow us to have scientific discussions and advices with them. There are, I think it was also mentioned in the previous deck that please have discussions, go for scientific advices, please have discussions with the agencies. And this allows them to be a part of the development, and there are no surprises. So when you go there and you discuss the data with them, they are not going to be surprised looking at the data because they are already aware of the kind of challenges that you have and what strategy you are using. It's a long process. During the review, there could be multiple questions. The final point is teamwork is the key to a good dossier. Everybody has to work together to write the story. It is one story, so it doesn't matter whether you are incorporating development data or quality data. It has to come out as one single story. So the clarity in the flow of information. There are different sections which carry similar information. So ensuring that we are not duplicating it or error, that one section carries a different kind of information, the other section carries another kind of information, and so on. So these are also very important points. So the last few slides that I have is representative data from Trastuzumab. So this was presented by the agency. They summarize the data once they are convinced that the data is good enough for an approval, then they summarize the data and they presented this data. So we had around 35 tests assessing all the quality attributes, many orthogonal methods, a lot of functional activity assays were included. So this is a list of that quality attributes. These are the number of lots that we had. So we had around 28 lots of the US Herceptin batches, 38 lots of the EU batches, and 16 of our own lots. And as I mentioned, statistical tools were important, or they were required for So there is a the assessment required of tiering. Again, all of this information is available with the US FDA. I think there are guidelines around it. This slide is talking about the data from the functional assays and the summary of the data by the FDA that all the assays are passing the equivalence testing. That's one part of the story, but the other part of the story is where there were differences. So if you can see here, there are three of those attributes like total mannose, the non-glycosylated heavy chain, and total sialic acid, and also the charge variants were found to be different. So there are differences. But those differences did not preclude that the molecule is highly similar. So this is a statement made by the FDA based on the data that we had provided to them. So they said that there are minor differences observed in the content of some species. However, I'll just go to the last slide. Yeah. So this is the slide which talks about how we address the differences, and they were convinced that these differences are not going to cause an impact on the molecule, the biosimilarity of the molecule. So because there were a lot of functional assays, in addition to the clinical trial data, obviously the clinical trial data was critical also, very, very critical, but the CMC supported the data that these, whatever differences, small differences were observed, were not critical in establishing that the molecule is biosimilar. So this is the last slide. Totality of the analytical similar data supports this conclusion. Thank you.
I'll just check the questions. So maybe you had a question.
Yeah.
I'll give you a mic.
You can ask questions to Naren also. He was with us all along this process.
So this question is for both of you. Is there, when selecting the methods, and you said that method has to be robust enough, when you're selecting a method, because okay, for trastuzumab, maybe you know what was done before for the innovator's molecule, but when you start building a biosimilar, you don't know what the innovator did at that point of time to prove the glycosylation pattern was this. So the selection of method, is there a my way or highway kind of tone from FDA that they have used? This is the US pharmacopoeia thing written, and you have to use this only, or you can have your own.
So there's no such requirement. The method development is actually left to the person who's making the molecule. So your company can develop your own methods. It is completely your development. What you do is analyze both molecules with the same method, right? So you're establishing the quality of your molecule in comparison to the innovator molecule using your method. So this method is actually, the innovator does not publish that method anywhere. So there is no way you can even know what the innovator is.
Exactly.
So the agency also doesn't know. So there's no requirement.
Thank you.
Can we pass on the mic?
To mention, I'll also share my email ID with them. So if there are any questions, you can please send me a mail. I'm more than happy to answer.
Hello. Yeah. Hello, ma'am. It was very nice lecture because I belong to the background molecular biology, so I understand so much. I don't know the rest of the audience also the same. I feel so very thankful for the lecture. So there are few questions because ma'am in lab I'm performing molecular biology and I'm doing a cloning experiment. So the monocloning, you said right, it is a huge problem because when we do cloning, when we are going with that same clone for the protein purification after the conforming sequencing, it means that clone is not producing the same protein which we have already we defined. So how in industry level you guys manage this thing? That's the one question. And another is like upstream and downstream process. Can you comment on that? So this is another one question.
So the first question is
monocloning, like at industry level, because it's a big level, that manufacturing, and in lab also because we are selecting one gene and with that gene we are doing the cloning, and then also in the protein purification, like 6 liter or 7 liter batch, we used to get the challenges like that protein is not getting here or protein may degrade. So at industry level, how the industry people manage that thing, I want to understand.
So we develop processes. Suppose if you are observing fragmentation, so you'll have to identify what is the root cause of the fragmentation, right? It may not be linked to the clone at all. It may be linked to the protease that is present in the clone, or some other issue. Maybe there are additives in the upstream process that is causing fragmentation, right? So first is identify what the root cause is. What is causing the fragmentation or whatever is causing the breakdown of the product. And then you can go back and correct it, because it is not necessarily linked to the clone every time, but it could be also linked to the clone. It could be also linked to the clone. It is a difficult process because unless you scale up, you may not observe the problem.
Yeah.
Right?
Yeah, it is happening because when we clone, that time means colonies will come, everything will be sounds good.
Yeah.
But when it comes to protein purification,
so we face exactly. I mean, in the industries we face exactly the same problem. I have a colleague from molecular biology who can relate to you. He'll say that we have given you a nice clone. What did you do to it? It's now not producing the same product, right? So it is a problem that we all face. I mean, we just have to work around it. So how we would do is we choose many clones, right? We choose up to from 20 to 40 clones, and then look for those which have minimal challenges. So the testing of the quality right in the beginning, but that I was mentioning, is to look for CSDS will tell you whether there is high content of fragmentation. SEC will tell you whether the aggregates are higher, and so on. So try to incorporate quality testing during clone development. So in one of my slides, I had that, right? The last two steps, right? So when you are expanding, yes, when you are expanding the clone, when you are at the shake flask level or the next level, incorporate quality testing. It is a difficult thing to do because you have to do it for 20 clones. You have to do a lot of testing, right? A lot of samples will be generated, but that helps you from going further and coming back to.
And then another question is orthogonal test that I heard first time. So can you please explain what it is?
Orthogonal tests are for the same attribute. You need to do many analytical tools. So, for example, HMWP. You need to use an SEC HPLC. You need to use AUC. I mean, there are techniques that are available for the same attribute. Glycan, for example. You can use more than one technique. So that is called as an orthogonal. For critical attributes, the agency is, it's just more convincing if you have the same attribute measured by different tools and they give similar results. That's called.
Thank you, ma'am. Thank you.
So there are a few questions in the chat box.
Yeah.
So first one is it compulsory to conduct in vivo toxicity or safety studies for biosimilars?
That was for the previous.
I think a previous one. And the next one is what differential pre-IND considerations for oral biologics? But if you want to quickly comment on any one of those, or no. So are there any other questions?
Yeah. I have one.
If you can.
She has the mic.
Yeah, let her go first. There was a mic. Okay.
Yeah, so you mentioned that
It's on.
Yeah. So, ma'am, you mentioned that the stability studies, pre-approval and post-approval are different. So, are there any major differences in the approval? Because for submitting, for getting approvals, we have to show them that this is the method that we are using and we'll be using henceforth. So, are there any major differences, or there are some minute?
I wouldn't say there are differences. So basically, you can reduce some of the testings. You can. I mean, there is a possibility that potentially you can reduce some of the tests in your protein stability. But it is the commitment that you give is more a protocol. I mean, the requirement is to give a protocol that you will adopt, not the differences in the test pre and post. It's mainly a commitment that the agency wants, that you're not going to stop any of those testings, or you're not going to reduce the number of time points. For example, now you have given 3, 6, and 12, but after the approval, you just decide that you're going to do only 6 and 12, or you're going to do only once a year. So those kind of things should not happen, right? So what they do is they take a commitment from you that even after your approval, you will continue with pursuits. So then there is a discussion whether that is acceptable or not.
My question is more related to the data, and you mentioned about the data management is particularly the documentation part is very, very important. So my question is, I have seen a lot of US European companies are using the paperless documentation.
Yes.
Still, I don't understand why India is behind in that. That is first question. Second thing, as the process says that R&D process development, then it goes to manufacturing or scalability. Why can't we use the similar sort of platform, because we are using the different instruments in the process like upstream, downstream. In that sense, why we have so many islands of automations rather than having one platform and one thing? Because I have seen one company wonderfully, that is in Iceland, that is Allotech. I would like to meet you complete automation. You mentioned about the labels, mismatching, reading differently. Everything is barcoding. Why can't we become so advanced and take the advantage of the technology?
I'll answer as I know, but I think I'm going to transfer this question to Naveen. So I think cost is, for me, what comes to my mind is cost. I mean, lab notebooks are expensive, right? E-notebooks. E-notebooks are pretty expensive to set up a system. More importantly, to maintain it, right? I mean, you have scientists who are freshers who come to the lab to work. You need to train them, ensure that they're able to adopt and follow those systems systematically, right? And it takes a lot of efforts to maintain. Am I right in saying that?
Yeah, I think there's no one answer. I think it's a combination of many things. One of which Jyoti mentioned, that these what I call laboratory management, information management systems are very expensive. The ones that are available from international sources, right? And there are now some smaller Indian companies who are making them, and I think if any country can do it, we should be able to have done. Yeah. If any country in the world can be digitized, it should be India. तुम लोग करो ना सब कुछ करो. Like, I think you hit the nail on the head, right? Efficiency can improve so dramatically if we are doing that, but there is no one answer. I think we all have to collectively introspect and figure that out, and each company has to figure that out for themselves first. And we hope, I'll put a pitch for this effort that Smita and the Venture Center is trying to put. If we all come together every month, we solve every problem.
So that's a problem statement for if anyone wants to work on that.
Yeah.
Affordable automation.
Affordable. Right. So I'm from the same domain, automation, what you are talking all about. And in-house, we have developed automatic formulation batching system that takes care of everything right from entry of the raw materials to deliveries going to the clients. And even it takes care of the development of trial formulation batches, its implementation, everything is barcoded, and the consumption is taken up from the system itself. You cannot say that suppose somebody has added 2 kg, 839 grams, and he says, I have added 3 kg as a consumption. Even that is not the case. So what I tried now, the gap or the scope to improvise is adding all those factors which affects your batch. It is not only the formulation or the process automation, but there needs a hand-in-hand approach from the product development team and the automation team. And even the softwares should be developed a step ahead now, so that the science people would be able to express their concerns, and the automation people would be able to follow that to mitigate those risks, and then the improvisation can happen, and would be happy to contribute on that.
I think you're already working on something.
I mean, we should definitely talk more.
Sure. Thank you.
Thank you.
Myself, Manika from VCCBA team. We work on biophysical characterization. There was a one line that sialic acid and other one glycan has impact on pharmacokinetics. Why only these two has a impact? Because there are other like N-acetyl glycosamine, galactose, glucose.
So in that particular product, differences were observed in those two attributes. So if the mention is that those two attributes, in spite of having a impact on pharmacokinetics, it does not impact the overall safety of efficacy. So,
does it have a more abundance on those molecules? Like
no, for the biosimilar that we were working on, those were found different. All the others were similar. Like N glycose, that was found to be similar. All of the other glycan modifications were similar. Only these two were different.
Okay. Thank you so much.
Thank you very much, Dr. Iyer, for sharing your experience and insights. I can see there are a few questions on regulatory aspects in the chat box. So those would be taken by Dr. Sami Sangeethra after his talk. So I'll now introduce our next speaker, Dr. Sami Sangeethra. He's Vice President, Head, Regulatory Affairs and R&D QA, Biologics at Zydus Bio. He has 24 years of experience in the leading biotech and pharmaceutical companies. He is skilled in strategic planning, project management, R&D QA, and regulatory affairs, domestic EMEA and regulated market. He is a strong healthcare professional with regulatory affairs certification from Regulatory Affairs Professional Society USA. He actively works on regulatory reforms and policy shaping and has represented ABLE, OPPI, IPA, FICI for several regulatory reforms in India. With this short introduction, I welcome you for your talk. Do you want column? Yashodhan, I hope the presentation is visible.
Yes.
Thank you, Smita, and thank you, Dr. Nareen and the Venture Center for inviting me here. Today's topic that I will be covering will be biologics regulation and biosimilar product development. So I will be covering biosimilar stages of development. So what are the product development stages and regulations of EMA, US FDA, and India. So we all are working in a regulatory industry, and when you are in regulatory industry, we have to follow, work is governed by rules and regulations. So can you name any regulatory industry that you are aware of, apart from pharma?
Medical devices.
Medical devices, banks, all this, whenever we put the stock market is regulated. So when you are having regulated market, you have to follow some rules and regulations. So we are in pharma industry. So we have to follow the acts, rules, schedule, guidelines, etc. This is very important. And even if you are doing startup, even if you are doing research, this has to be kept in mind, because all data that you generate is going to go to some submission one day or the other. So these are the stages of biosimilar development. So today's talk, I have put on only biosimilar, because then we can, and I will be throwing some information on the new novel biologics whenever we get that topic. So essentially, we have to identify a molecule, develop the clone for that, develop analytical methods, then develop the process for drug substance, that is upstream, downstream, drug product formulation, and then finalize those things. So it is very important that you finalize before you move ahead for the next. And I remember Stephen Covey's one rule, begin with the end in mind. This is very, very critical for biosimilars, because if you don't know your target molecule, if you don't know the characteristics of the target molecule, you will not be able to achieve that. So as Jyoti has already said, you have to analyze a lot of reference product to set your specifications. And that's the importance of quality by design approach. And analytical similarity is very key, and that should be done at all stages, including clone development, which Jyoti has already emphasized. And these stages are same in USFDA, EMA, and CDSO. So there's no differences in this stage. So even you are a small biotech company or a very large biotech company, the stages are same. Now, once you have developed the process, method and specification, then you move to the next stage, and that is taking consistency batches, whether you are sitting in US, Europe, so that you understand that yes, I have the process and product in control of my batch profile. And then you do analytical similarity, where you take several batches of your product and reference product and do analytical similarity. Then you do non-clinical similarity. However, the relevance of non-clinical similarity is going down day by day. And then for biosimilar molecule, you have to do a PKPD and a phase 3. Phase 2 study is not required. And then once you have all the analytical similarity, non-clinical and clinical similarity data available, you will be going for a MA submission, whether you are in USFDA, EMEA or series. And then you get authorization and you launch the product. Again, here CMC and GMP requirement is very critical. And I would like to again emphasize what Jyoti has said. GDP, that is good documentation practice, is very critical for all the stages, R&D as well as manufacturing. Validation of methods and process takes place in this stage 2, and pre-approval inspection is also there. If you look at from the point of view of advice, a lot of consultant can also advise you, but it is recommended that you take agency advice well on early, so that we can really develop the product based on the advice. Though a lot of information is already available on various biosimilars approved so far, but taking the advice on that particular molecule will be helping you to define what are the methods they are looking for. If you skip anything, they will surely add that. For US biologics product development type meetings, you can have and you can run the program to open that. And for EU, you can do QP audit, that is quality person audit. This is mandatory for clinical and also for MA. For MA, agency might also come for the audit. If you look at from the non-clinical point of view, if analytical similarity is well established as a highly biosimilar molecule, then that non-clinical studies can be viewed. And that's a good thing that now a lot of guidelines are talking about. But interesting thing is also that UK and WHO is also trying to say that now phase 3 also can be viewed. If you have a good analytical package and if you have a good PK or PD study similarity established along with immunogenicity and with immunogenicity assessment, you can also, UK and WHO draft guidelines says that you can also get waiver of phase 3. If you can justify properly, you can get that waiver. So biosimilar development basically takes there are 3 components: analytical similarity, non-clinical similarity, and clinical similarity, and we'll just go through briefly on that. Analytical similarity, Jyoti has already taken quite a deep dive there, so I will be just going through. Geochemical characteristics methods are required, biological characteristics are required, orthogonal assays are encouraged, several batches, more than 6. You already seen the figure 2838. So head-to-head comparison between the biosimilar and reference product. Region is very critical. Suppose you want to go for US, Europe, what biosimilar, what reference product you will be choosing is very critical. And it is important that you choose US and Europe both, do the analytical similarity and the PK study, so that you can do a phase 3 study only with one kind of region, and that is acceptable nowadays. Statistical comparison is critical. And the assays that you have you've seen in the Jyoti's slide is around 15 to 20 assays years old, but we have to do 30 to 35 assays head-to-head comparison to really say that yes, my molecule is highly biased. With respect to non-clinical similarity, in vitro studies are encouraged, where we can do a functional assay testing, receptor binding assays. Then once those tests are well established, you have to see if there is a residual risk, and then do a risk assessment. If there are at all some differences, then you should do a repeat dose toxicity study. Local tolerance can be part of it. And the consideration is that you should do the study in relevant species, and most of the maps relevant species is monkeys. The route of administration also should resemble the clinical use. If it is the IV drug, then you have to do the study in IV. If it is subcut, then you should do the dosing in the subcut in animals. comparative studies are mandatory. However, the relevance of toxicity studies is going down day by day, and that is due to the well characterized characterization of the product, and then you can justify that. Safety pharmacology, reproductive toxicology, and carcinogenicity studies are not required for biosimilars and biotech products. With respect to clinical similarity, a stepwise procedure that should begin with PK study to prove the similarity in PK, and then if PD is also available, and then the clinical efficacy and safety study will come. In PK, you can go with a single dose crossover study if it is a cytokine molecule. If it's a MAB kind of the molecule, then parallel studies are recommended because of long half-life and risk of immunogenicity. With respect to phase 3, you have to select a sensitive population. There are many indications a product is approved, so you have to select the indication which is more sensitive, homogeneous patient population, study to include safety, efficacy, and immunogenicity parameters. Clinical endpoints has to be defined, primary endpoint, secondary endpoint. Same RMP should be used throughout the clinical development, and this is sometimes important to note because a lot of RMP in other companies, they change their formulation or concentration, and you have to keep this point in mind. Interchangeable product, as we all know, Biocon's insulin is now approved as interchangeable molecule. So those kind of studies are required where you can do a crossover study and show that in the phase 3 that yes, my molecule is showing the same efficacy and safety study results. Extrapolation to other indication. So consider adalimumab where you have 9 indications. So you can't do studies in all the indications. So you can go, and agencies allow that if you do one study, you can extrapolate to other indications, provided biosimilarity is established with respect to analytical, non-clinical, and clinical, at least in one indication, and mechanism of action is same, and involved receptors are same. Also, if you look at from the immunogenicity point of view, from our US and Europe perspective, that also is addressed with respect to your safety concerns. Now, let's go to the agency and understand how the agency look at this. So that was common part for most of the agencies. So EU directions came in first introduced in 2001. Basically, 2001 was when India's first biosimilar was launched, erythropoietin from Opioid. And in 2003 was incorporated as a directive for marketing authorization and preparation of biosimilar medicine product. In 2005, they released the overarching guideline, and in 2006, they released the non-clinical, clinical, and quality issues guideline. And the first molecule to be approved in Europe was Omnitrop in 2006. These were the 3 overarching guidelines, which talks about the licensure issue in the first, second, non-clinical, clinical, and third, quality issues. So these are basically what are they expecting in non-clinical and clinical, what are they expecting in quality, like they're talking from expression, manufacturing, reference standard, RMP, all those kind of issues they're talking about in these guidelines. Then they quickly understood that all the companies are coming to them and asking for advice. So why can't we have product-specific guidelines? So Ima has given a lot of product-specific guidelines, and they have mentioned exactly what non-clinical and clinical data they are looking for in the biosimilar applications. They also have a monoclonal antibody product-specific guideline. And it is very good to follow this because then your scientific advice package, whatever questions you want to ask, are very clear, and you get a proper advice. Then they also have adopted recently the Q5E, which is the process comparability. Then they also have comparability for non-clinical clinical issues. Then immunogenicity assessment is a hot topic now, and a lot of guidelines are being published for that. And also pharmacokinetic considerations. So these are good guidelines. If you are developing a biosimilar molecule, you have to basically know each guideline in depth. Now, coming to advice. Now, this advice could be used for novel biologics as well as for biosimilars. So you can, once you are ready with the package, you have to prepare a package which has a meeting request, meeting package, a lot of questions, a lot of introduction of the molecule questions, as well as with respect to company position also you have to give for each question. So after submitting, they will have a meeting, and then after 30 days, they will have another meeting for report discussion, and joint report will be published in day 40, that is internal publication, and they will have meeting with company on day 60, and day 70 they can use the full advice. So this is the scientific advice I will cover in the CDS. So India also has in 2019 started a pre-submission advice, which is very good for all the startups and who are developing novel. It is good to go to Indian authorities also, learn about, tell them about the molecule and learn about the requirement of clinical, non-clinical. This is the Ima Regulatory Procedure. So once you've done all the studies, you will be submitting, you will be doing a pre-submission meeting with them so that you understand the dossier issues or anything that they are looking for. Then you will do the submission. They will start at the day 0, D0 is day 0, and the report will be given at D80, that is 80 days. Basically, it is a validation of your dossier. After that, on day 120, they will give a list of queries, and the clock stops there. We will have to respond to queries, and then after the response is submitted to the agency, they will open the clock, and the day 121 review starts. They will have the joint assessment report by day 150, and later list of outstanding issues on day 180. Once those are responded successfully, they will give positive opinion, and then EC, and then the European Public Assessment Report will be published. So this is a very straightforward and timeline also is well managed by the EMEA for this. With respect to US FDA, the BPCA Act was published in 2020. During this time, if you look at the Indian approvals, India must have approved 40 to 50 products in India as biosimilars. And actually, the act was acted on in 2010, first introduced in 2009. This creates an abbreviated licensure pathway for biologic products and a license under 351K. The novel biologics is licensed under 351A. So these are the guidelines. So if you see the guideline 2015, though the act was passed, but there was still a lot of litigation, a lot of discussions with all the innovator companies, biosimilar companies, associations, and then they finally published the guideline in 2015. And I'm sure 60, 70 molecules would have been launched by that time in India. So this scientific consideration and quality consideration are the two key guidelines for going through when you want to develop a biosimilar molecule. Scientific consideration talks about not only analytical similarity, but also non-clinical and clinical similarity. Quality consideration talks mainly on the analytical similarity package and what is required in that. Then there are also, like EMA scientific advice, FDA has pre-IND meetings, IND and pre-IND meetings, while in case of biosimilars, they have shifted the from pre-IND meetings and they have a PPD meeting. So we'll talk about that. They also published a lot of interchangeable guidelines and other Q&A for explaining things. And these are the scientific advice, or you can say BPD meetings. So first is a BIOM meeting. This is biosimilar initial advisory meeting. This is when you want to understand whether my product will fall under 351K. So that is the main purpose of this. So you go to the agency with preliminary analytical similarity and then inform them. And within 75 days, you'll get a meeting. And what happens there is that on 75th day or 74th day, you will get a response saying that these are the queries they have, or this is the agency consideration, agency opinion. And then based on that, in the next day, you have to explain your case or agree with the agency. So that happens on the 75th day, and then another 15 days, you will get a formal response from FDA. and that first meeting is free. And then thereafter, the meetings are very, very expensive. You have to pay initial fees as well as annual fees. Type 1 meeting is granted very quickly in 30 days because it is on the clinical hold. Suppose you have taken the advice and you have started a clinical trial and there is some safety concern, etc. And if you immediately go to FDA, that meeting is granted in 30 days. In type 2 meeting, you can discuss the design, etc., and also some of the analytical similarity data that is granted in 90 days. If you want in-depth analysis, so they will also require more people to review. So that's why the days are more, 120 calendar days. And this is basically when you have you're very close to submission and you go to them with all the package to understand what are the still open areas and you want to work on it. And the last type 4 meeting is basically a pre-submission meeting, which is basically to understand the CTD issues if there are any concerns there. So that meeting is granted in 60 days. But once you open a BPD program, you have to give the fees. Here, when you do a submission, you have to give meeting request and meeting package. Meeting package consists of molecular information, questions, company position. That okay, this is my question. Okay, I will be using 15 batches for RMP, etc. And then we have to justify that what is my company position about it, and then agency will opine on that and give some background about why you are saying 15 numbers is correct. And this is the USFDA review process. So we can again have a pre-submission meeting with them. Review starts at day 0 instead of EMA day 80. Here at day 74, where they give filing review issues if there are any issues. And then 5 month and 8 month is mid-cycle and late cycle review. And you will get some IR. IR is information request. So if they feel anything incomplete or if they have any query, clarification, etc., they will send a IR information request notice, and then you have to submit that information. If there is major amendment required, if they ask you to do some study, etc., then there's a 3-month extension you can take. Once you have submitted everything, they will issue an action letter on 10th month and the approval or complete response. Parallel to this, there will be a inspection by FDA of your facility. If you're lucky, you don't get any 483, you can good to go. USFDA uses approaches like stepwise. So stepwise is analytical similarity, non-clinical similarity, PK similarity, and clinical similarity. And they also look at totality of evidence. So we have seen in Jyoti's presentation, there were some issues with respect to charge variant or some glycan. So and they have looked into as totality that it has not impacted the PK, it has not impacted the safety, it has not impacted the efficacy. And that's why in the totality of evidence, still there are some differences. They have approved the product, which is a very good actually approach. Everybody must have heard about orange books, so I wanted to also we Biologics have purple book now, which lists the licensed biological product and reference product exclusivity, as well as patent information, and all those information is available. It's good to go and look at those purple book. Now coming back to India, Indian authority is CDSCO, Central Drugs Standard of Control Organization. It's in Delhi. So to give brief about the regulatory bodies, DCGI is coming under Ministry of Health and Family Welfare, and NIB Noida is also under MOHF, and IPC Ghaziabad. So what does DCGI do? They monitor safety and efficacy, approval of clinical and marketing authorization of the drug, and post-approval changes also DCGI for biologic they do. And NIB Noida is responsible for biologic product testing. So if you are launching a new product or first biosimilar in India, then you have to go to NIB for testing that. IPC Ghaziabad is responsible for establishment of monographs for biosimilars and biologic products. RCGM comes under Ministry of Science and Technology, and they are responsible for monitoring the cloning development. So the IBSC and RCGM, they give approvals for import of clone, import of gene, etc. And they also give approval for preclinical studies. So basically, they look into the analytical similarity package, and then they give approval for tox studies. And this again will be, they also will be looking at the tox results before they can give a NOC for going ahead for a DCGI for clinical trial. Then we also have a state FDA, which comes under state Ministry of Health and Family Welfare. They grants manufacturing license. They are responsible for WHO GMP certificates, UPP, audits of plan, etc. So there is no single window like FDA in US. We have to approach so many agencies. And since 2008, I would say that Indian authorities are publishing a lot of good guidelines, and I wanted to show this today. They have a CDSO guidance for industry for biologic product, where they have clearly mentioned they have earlier there was Form 44, which was information required for drugs, so that was same used for biologics. But now we have a CTD format incorporated for both clinical and non-clinical new drug approval, that is the BLA. And post-approval change guidelines is very much published now in India since 2008 and followed. And preparation of quality information for submission, that is also we will describe what are they expecting. Then there is also in 2011 guideline on IBSC published by RCGM and DBT, and that's also quite a good guideline. In 2012, before that, there were 2-3 guidelines which were talking, but nothing was in one guideline as a biosimilar pathway or similar biologics pathway. So this is the time 2010-11 when DCGI constituted a task force. I was lucky to be part of it, and they said that okay, we don't want to call biosimilar because in US it is still not finalized what they want to say with the follow-on biologics, biosimilar, similar biologics. So India decided that we'll call them similar biologics because we also wanted to put vaccines under the similar biologics term. So this guideline was published after 4 years. A revision of that guideline was published, and the key differences were acceptance limits should be set based on reference standard. This line was added. CQA and KQA was added to it that you have to specify in your application. Then abbreviated comparability will no longer be acceptable. So they also wanted to, because by that time a good number of guidelines were published on how analytical similarity should be done. So they wanted a complete package. Phase 3 requirement, which was not defined as a number. So they said that we want at least 100 patients on the test arm and mandatory phase 4 of 200 patients. So did that to include the safety aspect of it. So this was quite a welcome, and this was well appreciated worldwide that India has now come up with a guideline. Then, as we all talk about digital world, Sugam started an online portal for submission for biologics in 2017. And in 2019, new drugs and city rules was published. Here, the key items introduced were pre-submission and post-submission meetings, pre-approval and post-trial access, which was a very important thing for the cancer patients. Time-bound approvals for clinical trials, so that was also published. More reporting of clinical trials quarterly and half-yearly, so that no adverse event or safety is missed. And clarity on phase 4 and post-marketing studies was provided. With respect to Indian regulatory process, again, the stage 2 remains same here. The only thing is I've added the agencies that were involved. So after analytical similarity is done for non-clinical protocol approval, we have to go to RCGM through IBSC and then RCGM and get the protocol approved. After the protocol is approved, we have to go again to IBSC and RCGM, show them the result that it is comparable. And then parallelly, we can also submit to SEC and to DCGI actually. And then we have a SEC meeting and DCGI approves the product for clinical trial. And then after the clinical trial, we have to go to DCGI again for the MA. And then the SEC is a subject expert committee where you have doctors, academicians, and they look into your clinical safety, efficacy, immunogenicity data, and based on that, they recommend DCGI for the approval, and final approval is coming from DCGI. Then once we get MA submitted and MA approved, we also have to go for joint inspection. And state FDA is one of the key agency here who conducts the joint inspection. And for the joint inspection, NIB as well as DCGI people are also there as a joint inspection. And then we get a manufacturing license, and then we can launch. These are the days provided by the agency as working days for the individual application. Though if the queries are more, the days, the deadline is not followed. Now let me take you through the current situations in US and India. If you look at from the European perspective, there are 67 products approved so far. In US, 35, and in India, 80 plus products are approved. These are multiple brands of individual molecules. Like for example, EPO, we have 5 brands. Adalimumab, we have 4 brands. Trastuzumab, 3 brands. So 3 companies manufacturing the same product. Biosimilars have a lot of hurdles. The first hurdle was scientific. Everybody thought that it can't be manufactured. As a scientific, nobody can manufacture. So that has EU, US, and India, that has been hurdle is crossed. Parliament, there was in US, they said that, oh, there's no act. How can you develop in 2010, the act was enacted. Then they said that, okay, there's no regulation. You have act, but there's no regulations. In 2015, the regulations were there. Then there's patent dance after even several years now. Amgen is still not able to sell Adalimumab. They're still in litigation. Then there are physicians who say that biosimilar has done less studies, and that's true because I was with Roche also, and Roche, I see that for each molecule they have done 16, 17 studies. For each indication, they have 2 to 3 studies. So when you look at the biosimilars, they do a good TK-PD study, or PK in most of the molecule PD is not there, and a phase 3 study, or maybe an immunogenicity study, 2 or 3 studies. So the physician have their own concerns, and I think partially it is one, but still a lot of miles to go. The only party which is very happy is insurance companies because they get a cheaper product. Then the pharmacist, if you see now, if doctor writes trastuzumab, they don't know whether to prescribe Herceptin or I forgot the name, but Mylan product. So it is that duality. But now with insulin, they also don't have that because it is interchangeable. So that battle is also slowly crossed. And the last is patient. Now, once you're a patient, you say that, okay, 16 studies, 17 studies versus 3 studies, what to do? So that is where India has gone ahead with respect to total affordability. The product is already gone through regulations, and it has been used by all the premium institutes, to say hospitals. And that's how the patient also convinced that, yes, the drug is approved, gone through all the stages of development, it can be taken. So few hurdles crossed, still few to be crossed. And these are not the hurdles only. There are other hurdles. Innovator companies drop prices. So that is a big hurdle. Second hurdle is biosimilar companies. You are not the only company selling the product. So there are multiple biosimilar companies who are going to be targeting that. And so going there first is very important. Second generations and improved versions. So, for example, if you go with trastuzumab, now there's Kadcyla. And now when people developing Kadcyla, there's Enhertu. So there's always that each companies and their improved versions will be coming to fight the same market. There are also new chemical molecules targeted for the same indication. So that those hurdles are there. But one good thing is that the insurance payer is there. There are regulations, scientific literature is available that yes, biosimilars can be developed, manufactured, and there are no side effects, adverse events, etc. So based on that, the biosimilar companies, and if you're working biosimilars, you have a great future because there are many, many molecules in the pipeline that you have to develop a biosimilar. With that, as per request of Smita, I'm also talking about Alcoa. And this is very, very critical because once you develop, even if you are doing you are in a venture center, you are doing something for the market. You are doing something so that you earn, right? Can I get yes for that?
Yeah.
Everybody. So if you want to earn, you will have to give documents to somebody to get the approval. So if you want to give approvals, and take an example that you have done all those good things, but you missed on one document and your product is delayed by 6 months. 6 months is 6 months revenue. So for you and also for the company, you are lost that.
Yeah.
So it is so important. I will like to go through the attributable is that whatever, if this document is there, it says that BioRac Venture Center, that is this. But in other ways, if you are in the lab, you have to sign it and date it. And backdate is not allowed. Okay. The second thing is legible. The data should be readable. Otherwise, if it is like 2% or 24 or 27, as Jyoti said, so it is very important that it should be legible. And this, according to me, I was talking with Smita that we have to tell this to our education minister that this should be taught in schools. Then contemporaneous is very important because we have a limited memory and our memory is hijacked by our social media also now. So you will not remember what you have done yesterday. So it is important that you write today only so that tomorrow is no problem for you. Original, like for example, everybody might have a lab notebook here, right? Okay. So how many times people have written something on tissue paper? Because I was there, but a lab notebook I was not able to see. So that thing happens. So what we have done is we have removed pencil. So we removed pencil, tissue papers, rubbers, sharpeners, as well as sticky notes from the labs. Because otherwise it is not possible. If it is there, you will use it. If pencil is not there, you can't use it. So just remove it. We also requesting HRs that they should not give pencils and rubbers in the kit. Okay. So that's contemporaneous. Original is when you say that this is the lab notebook. I can say that okay, I photocopied it and for this only experiment I'm using it. So that is not a good idea. Original is original. If I show to the auditor that this was done, you will say okay, which is the notebook issued to you. So that if you are only 3 notebooks, 4th notebook is not yours. You might have signed it, but it's not original. Data should be accurate. If it is, suppose you are referring to a temperature 25.7 degrees Celsius, 25.8 degrees. Suppose you in hurry and you said 25, miss the decimal, then that is important to also make note of. Like pH is very important that you have the complete decimal points. Complete. Now, if I say to you that okay, you have a full report, but you have missed the raw data there, so it's not complete in that case because you are just assuming you have prepared a report without data. Consistent, you have to be consistent with respect to your data is consistent. It is not there are no gaps in that data. Enduring, that it is not like thermal paper. A lot of people use pH thermal paper, right? So if it is thermal paper, it should be photocopied and put in your journal or log notebook. Otherwise, you will face problems in the audit because the raw data is not there. If raw data is not there, you have not done the experiment. Okay. And available. So available means that if I ask somebody, okay, show me the raw data of this, and that person says, "Dikhta hu," well, it's not available actually. That person is just saying that it is there, but it is either destroyed because of some issues, or that experiment was not done, it's written. So that's the interpretation. So it is important that you understand these things, and I would recommend, I can share this slide. You just take pictures of this slide, colored, and post it in your lab 2-3 places. It is so important. Thank you.
Yeah, thank you very much. We can take up questions. Yeah, you can keep this. I think on my.
Yeah, thank you for the presentation. So I had a couple of naive questions. First is getting an approval from, say, US FDA or EMA. Does it guarantee approval from other subset of countries? You can directly get into them, or how?
Very good question. Actually, if you get a letter which says either US or Europe, in at least 15 to 20 countries, you can just walk. Okay. Very easy. Okay. So a lot of people say that they have a strategy where India first and then they go for global. I think the overall strategy is now reverse. First go to US, other countries are very easy.
Thanks. The other question is, how transparent is the process in Indian setup, like the CDST?
Indian process is very transparent. They have guidelines, they have all the information. It again depends on how much data and how much clear you are writing your dossier. So if the dossier is not written correctly, there will be questions, and how fast you can answer the question, that's how then the deadline will not be met.
Thank you.
So even I have a couple of questions.
Yes.
My first question is, considering the classic example that we had recently because of pandemic, COVID-19 vaccine. So do you think that the timelines will be improvised, considering that as a control?
Okay, so I would like to, I didn't have the slide yesterday for this presentation, but yesterday I was going through my earlier slides and I saw that slide, and that reminds me when you asked that question. Actually, if you look at both industry and regulators and academicians, everybody was fighting for growth. Okay, there were a lot of concessions given for COVID by regulators. They allowed stockpiling, which is not a norm. They allowed, they took SEC meetings every week for COVID, which is unlikely. Like I used to submit earlier and get the SEC meeting after 2 months. So they have done marvelous thing, and not much appreciation has been done for the regulators for that, but I feel that they have done a marvelous job, as well as industry. Like you see Serum, you see Bharat Biotech, Zycovid, which is our product. It has been like developed and in the market in like 2 to 2.5 years, which is very, very remarkable. And that's a very good thing for us, that if we are challenged, we can work. The only thing is, can we self-challenge?
My other question is, you showed the ALCOA principles right now. So will automating entire labs, like end-to-end systems, help to achieve ALCOA very easy?
Okay, I was wanting to answer his question that time, but data integrity or documentation is not soft or hard issue. It is mindset issue. If you have a mindset, like for example, you must have heard this many times, that if you are in US, you will not throw anything outside. That's a mindset issue. What happens when you land in US and not throw the paper while you do that in India? So that's a mindset issue. If you are, like, there are people, I have I'd like to name that person, Abhijar, who is, I think, with Enzyme now. Very good documentation. Like, you will love to read his lab notebook. So meticulous, so nicely used to write. So those examples has to be highlighted, shown, and like when he was a protein purification scientist, when he was sitting in front of ICTA or the protein purification system, you can't do anything. The system is running itself. So you can take notes there, like HPLC. HPLC is running itself. You don't have to do anything. You prepare sample, do the documentation properly, and then when you feed, you have enough time to think what's happening, what's to be done next, etc. So that time utilization, I think one good thing that you can do is keep your mobile aside. You can do a lot of things.
Thank you. Thank you, sir. It's a very nice lecture. So I agree with you. Even gel also take 40 minutes to learn, and PCR also take 3 hours to learn.
And then what happens is you prepare the sample.
Yeah.
That time you say, I'm preparing sample. How can I document? Okay. You can't document.
But my boss asked to be a plus.
No,
first
you
write
can have
notebook
a
and then
you can have a template and then you can tick etc. That you can do. But normally what analysis does is that I'm preparing. How can I do documentation? So, okay, now the gel is running. Now you can do documentation. So that is a mindset issue. I think everybody has to train themselves for proper documentation.
Yes. So I have 2-3 questions to ask. So the first one I'll go with. That's why non-clinical similarity is going down these days. I want you to comment.
Yep.
And the PK similarity, because some of means here, so many are the industrialists, but yeah, students also here. So some words that we heard first time. So that I want.
Yeah, so if you see the non-clinical, when you're putting any drug in the animals will respond. The animal immune system, all systems will respond. But to capture the difference between two molecules, it will take a huge population of animals to really understand that, yes, that is the difference. While there are now sophisticated analytical tools that you can find those differences. And because of that, the relevance of animals is going down. Because they can't really help us giving that differences, because USFD or EMEA is what they are looking at, residual uncertainty. If that is addressed very well, then that analytical similarity is addressed. Now, with respect to PK is very critical. So anything can be waived, PK will not be waived. Because PK will tell you whether the product is in the system properly, because then only it can do the efficacy part properly. If there are some issues, binding issues, if there are excretion fast, then it's a problem.
Yeah, and sir, in your presentation, we saw that at one standpoint, biosimilar Indian status is 80 plus. So can you comment that, because it is very low?
It must be 95 plus right now, because this I wanted to go right, and that was 2019 public data of Government of India, which was 80. So, but I know that because 2 or more molecules not listed there, so it is 80 plus. I would say we should be very close to century.
And like well characterization leads the least residual risk. So do I agree with that? And is that necessary that all industries means first regulate that well characterization is must?
Yeah. So what happens is everybody is in rush to get the money from the market. So that is where you have to, industrialists have to have patience that yes, develop the product good, and then you can have that. So if that is done well, you have less questions to address from not only India, but also from emerging markets, US, Europe. So whenever you submit a dossier, if you're getting questions, that means your analytical package or process or methods are not proper.
So this question can be solved because if more PhD students get enrolled in research and they do sort of research, because they do characterization in their academics only, then so many molecules can deposit on the NCBI platforms and we can easily information and industries will help. So this can be solved by this. And the last question, can you just differentiate biologics and the biosimilar? What is the core difference between them?
Okay,
That's
so
the last question.
when you are developing a novel kind of biotech molecule, it is called NBE, novel biologics entity. When for that, suppose you have developed a novel biologic molecule and launched in US, Europe, India, etc. Now I want to copy your molecule, then I will call my molecule as biosimilar molecule.
Okay. Yeah. Thank you. Thank you so much.
Any other questions?
There are a lot of questions on regulatory.
Can you please elaborate on interchangeable?
Very good question. Basically, when you say that my product is interchangeable, you have to establish that even if, like for example, today you are taking one medicine, okay, company A, and tomorrow the biosimilar is company B. Suppose I shift you as a doctor, shift you from company A to company B. You should not get any adverse event. Your efficacy should be same. Because take an example of insulin in Biocon. So when a diabetic patient takes insulin, you are expecting that the glucose levels will be same by company A as well as B. So interchangeable means a product which can be really substitutable or interchangeable in that function. And there is no impact on the patient. It will have the same efficacy and same safety.
Thank you.
First of all, thank you, sir, for a wonderful lecture. My question is that if we are preparing a new formulation like tablet, then we are using a placebo, which is prepared from sugar molecules now. But in case of similar biosimilars or biologics, is there any placebo? I'm curious to know that what is the material which is used as a placebo in case of biologics and biologics.
Correct. So when you're developing a new formulation, if you have some worries about the toxicity, etc., that time you can use a placebo, or normally it is called control. But when you're developing a biosimilar molecule, you want to show similarity to something, right? And placebo can't be that similar. Of course, placebo may also have an effect, but that's not the thing we are discussing here. So we have to show that suppose you have Apple and you have Samsung. Okay, so you have to show that both of them are similar phones. Okay, so in what way you will show similar is that biosimilarity. If you take placebo, it will not work.
So in case of biologics, new biologics.
Placebo is again used there. The comparable arm is the standard of care. Suppose I am using Kadcyla right now, okay, and as a drug, and I want to develop better than Kadcyla. Okay, then the standard of care is Kadcyla. Then I have to use Kadcyla as a comparative study for proving that my product is better or equivalent to that drug.
Thank you, sir. Samir, any example of where a phase 3 waiver has been granted because EU regulations have been talking about based on a PK/PD, and if you can prove the residual, but any example, because same has been incorporated in the Indian guidelines also. But I am not sure if there is.
In India, phase 3 waiver has not been granted as of my information.
Yes,
but in Europe as well as US, phase 3 waiver has been granted for GCSF, PEG-GCSF, PTH. Those three molecules where you have
based on the PK/PD.
Yes, because in those molecules you have PK as well as PD. So PD can be surrogate to your efficacy.
For your efficacy.
But UK has UK guideline in 2019 says that even for MAPS they are open if you can prove that there is no immunogenicity risk and your PK is very comparable.
So you had a first talk in terms of the insulin.
Correct.
For the insulin when they talked about it. But
yeah.
I don't think there were any examples.
Correct.
Okay, my question is about as biologics means the similar terminology having vaccines for biosimilars. So, what are the Indian regulations, particularly on the cell therapy, gene therapy, even the stem cell therapy is concerned? Even though you can add on with the rare disease of the heart drugs in that sense.
So I'm not an expert on those things, but I know that there are guidelines published by Indian authorities now for gene therapies, stem cells, etc. And I would recommend reading that. I'm not like my company is not working on those. So sure. But you can Google it, you'll get all the.
Yeah, but thing is that means whatever I know, means my knowledge is also limited in that. But particularly the stem cell, the stem cells it covers most of the thing. But when therapy comes, I think that time it was not much information. Probably the new regulation thing, I don't know.
I would like in the 24 years of experience, I would say that once the Indian government is near to guideline or publishes guideline, there are a lot of therapies coming. So I would say that if you see the Indian biosimilar guidelines, which was published in 2012 and 16, and by that time 50 products were already launched. So now India is taking the things ahead. Before launching the product, they are having the guideline. So I would say that, and I know there are some colleagues who are working in stem cells, and there are a lot of products being developed, not just like a concept.
Okay, in biosimilars, even though
I think CAR-T cell, you are also working there.
Yeah. So in particularly, I heard the word traceability. So how regulatory is connected with that, and how that operation works?
Okay,
from the clone level to the final product.
Yeah, so traceability as just a word, it means that whatever is done, are you able to get documentary evidence, raw data for that? Traceable. Okay. Now, when you say from the whole product perspective, it starts from whether I have used GMP cell line for this batch, which has gone to clinical. So it can go to all levels, whether I have done proper in process, DS release, DP release, all whether it's traceable. Whether my method was same or changed, that could be also a traceability. So everything will come as a, if you broaden your scope.
Yeah. Lots of questions on
There
chat.
are lots of
Yeah.
questions on chat.
It was interesting. Yes, what difficult.
So how do you want to do it?
I need to come here.
Yeah. I'll just move the camera here so people can see it. Yeah, please go ahead.
The CDSO has provision to reduce non-clinical studies or waive local phase 3. So far as CDSO doesn't look into non-clinical, it's the RCGM. So RCGM so far has not taken into, but maybe industry bodies have us to show the UK, WHO, US, and Europe guideline and try to convince RCGM. Or waive local phase 3. Local phase 3, India's right now is not waiving because all these molecules, first generation molecules like GCSF, PEG-GCSF, and PTH, insulin, they are already in the market. But maybe the new entrant can try that, showing the US, Europe guidelines that get a waiver there. Are there any plans by the agency to revise the biosimilar guideline? Yes, they are working on it. For MAPS, most relevant non-clinical species is yes, Cynomolgus monkey, do CDSO expert studies. Okay, CDSO is very liberal in this. Indian guideline says wherever possible, you should use that, and wherever not possible, you can use the Schedule Y, which talks about 2 species, and that is you can have rat and rabbit. Recently, submission of anti-BDNO developed solely in China was rejected by FDA. Would India regulator approve such product? I can't comment on that. As you must be aware, yeah. So there are a lot of PD-L1, PD-1 molecules under development in India. So I think Indian companies will surely go to DCA for that. How much time it will take for biosimilar from development to market? 4 to 5 years, around 6 months for submission and ML, that is manufacturing license, 1.5 years for clinical trial, so that's 2 years. Non-clinical approval and non-clinical is 1 year, so 3 years and 2 years for development.
There
Is that all?
No, there are more items. Not able to scroll. It's okay, maybe we'll take those later. Yeah.
Thank you very much. I would now request Dr. Srikanth to kindly introduce all of us to CBA facility. So Dr. Srikanth Raudri has recently joined Venture Center as a Chief Scientific and Business Officer. He has over 20 years of pharma R&D experience in leadership roles in the USA and India, Vertex, Readiness, SciLife, and now Venture Center. He has a successful track record of establishing state-of-the-art labs and high-performing teams and leading multiple cross-functional drug discovery efforts, some of which have reached clinical development and approval stages in diverse therapeutic areas. His experience includes both small molecules and large molecule biotherapeutics. During his tenure, he worked in innovator as well as contract research organizations where he collaborated effectively with many pharma, biotech, and academia partners across the globe. He has received his PhD from CCMB, postdoctoral training from Harvard Medical School, and business management certificate from Northeastern University. So with this brief introduction, I welcome you. And let me just share your presentation. No, I share it. Yashodhan, can you see the screen? Full screen?
No, I cannot see the full screen. I can just see a slide and also some more slides. Can you please go full screen?
So I think I'm facing some problem. Can we go ahead with this? Yeah. Is it now? Hello?
Hello? No, it's not full screen yet.
But can we manage with this?
Yes.
Okay, thank you.
Yeah. Thank you, Smita. Can you hear me? Yeah, I changed to this one because it was upset and crying over there. Nobody was taking. So everybody prefers this one. Okay, good afternoon, good evening. So the last presentation for today. Warm afternoon and AC is blowing. I hope still are awake. Thanks to Sami, created a lot of discussion and some laughs. So hopefully you are awake. I promise I'll be very brief. Whole intention of my presentation is to introduce our center to you and make yourself aware of this facility to use yourself or anybody in your circle. So as the end of the presentation, you will realize it's a very valuable resource to use. So what I'll do today is walk you through what is introduction to CBA, who we are, what we do, and a little background for that. And thanks to Jyoti and Sami, a lot of the background they covered. So initially I thought I'll put 4 or 5 slides. I think my intuition works. We were going to present that one. So I'll be very brief with that as well. So some of the questions were answered. What is a biologic? What is biosimilar? So that those students who are still want to see in writing, so this is where it is. Biologic drug, as they already described by Jyoti and Samir, is a large complex molecules made from living organisms. It also involves very complex process to develop this. Biosimilar is a copycat of biologic. So we are very familiar with small molecules. The one on the left side, where we use a lot of generics in everyday life without realizing that it's a generic. But they are very simple to do. You can 100% reproduce them. Whereas in case of biologics being complex and produced by living organisms, you cannot 100% reproduce them. That's why Jyoti and Samir went to pains to make us aware how much complex it is and what all the things involved in this process. So having that in mind, again, this slide is introducing to where CBA comes into picture. Okay? So this is a very popular figure where we have two pyramids, one inverted over each other. So on the one on the black and gray is a development path, also called 751A, right? Where you use extensive development. In that case, as you can see on the left side, on the right side, analytical, preclinical, PKPD. As you go along the development path, the complexity increases, the cost of the development increases, the time increases. So that's for the originator development. Whereas in case of biosimilar, which is at the bottom, and which is in blue color, is a bottom of pyramid. Basically, lot of effort goes in the bottom. That's analytical side. So that a lot of that again is presented by Jyoti and Samir, and why it is important to take care of this part to get the approval faster. So this part, which is not trivial, it involves very resource intensive and involves multiple methods and lot of expertise, trained people to take care of this analytical characterization. So this is where we come in, CBA Adventure Center. So just go to the next level. So what is CBA? CBA is Center for Biopharma Analysis. Basically, it is hosted by a venture center. So they,
Hello?
Yeah.
Shrikant sir, sorry to interrupt you, but we cannot see the slides moving.
You can't see the CBA introduction slide?
No, we were not able to see the slides moving.
Okay.
Just it was fixated on only one slide.
Okay, okay. Give us 2 minutes. Are the slides moving now? Yashodan?
Yes, they are moving.
Okay, thank you very much.
Okay, so this part of analytical characterization, so as you know that Samir presented, India approved lots of biologics and around the world there are many more. So this characterization usually done by the big pharma within their organizations, and there are some big CROs where typically not accessible by small players because of the cost or the business model of those CROs, which are oriented towards West or developed markets. So small players, small companies, academic labs have difficulty in accessing these facilities. So with this in mind, Venture Center, with the grants from National BioPharma Mission, BIRAC, envision to have a center like this within the Venture Center to take care of that need or gap in the service. So the CBA is main objective is to become one-stop destination for quality in vitro characterization, and also it's open access. As I said, all the CROs, big pharma, are not accessible for everybody. So this will be a facility open for everybody. And also we have applied for GLP facility, so certification. So it will become a GLP compliant facility, even though right now we don't have it, we already use those practices. And where people don't have expertise, we can also become like Venture Center does with all the startups, we can do hand-holding and advisory role as well. So where are we now? So we have a fully established facility. We have a team in place, very dedicated team. So this team involves technical, quality assurance, and also we pay a lot of attention to EHS. Collectively, we are, as a team, we have 50+ years of experience in this area. So we have the team that is over there, a lot of faces you can see in the audience. So you can interact with them during the dinner time. And also facilities, as we speak, it's across the wall. So if you're Harry Potter, actually you can cross through. But only thing you cannot see Hogwarts there, you will see something like Matrix, strange looking instruments. We already working with many partners. That includes academia, small startups, and also big pharma. So what do we do? What all services we offer? So there are two boxes here. Left side is structural analysis. Right side is functional analysis. These are typically what we do. Most of the biologics, biosimilar companies do. We have a host of these services already in place. We have generated a lot of case studies, working with some of them internal, some with the partners. This involves, typically speaking, primary, secondary, higher order structure analysis. We also have SPR to check the binding analysis on the structural analysis side. And the functional analysis, again, we have a battery of tests we can do. We have all the expertise as well as instrumentation to do those things. For more information, again, you can visit bioanalysis.com. So for scientists, I think seeing is believing. So I have some glimpses of data that we have produced in-house. This is functional data. So on the left side, we have the cell-based assay for ADCC using CD20 antibody. On the right side, we have ELISA biochemical assay data. This is for TNF alpha binding. And then next few slides show the structural analysis. This is for bevacizumab. So this is you can see the peptide mapping after trypsin digestion. Then you can see there's 100% sequence coverage. So the same represented in a different primary sequence manner to show that the 100% sequence is covered. This is for intact mass analysis. Again, disulfide analysis, the same thing. So basically, just a glimpses of data that we have. There's a lot more data that we can share on case-by-case basis. If you need more information, this is the people you can contact, or directly go to bioanalysis and put in your query. We're happy to help. As I said, take advantage of this facility. We are not money-making institution. We are here to help. Thank you.
Thank you very much, Dr. Ima. And I request the audience that you can interact with our team members later during the networking and dinner session to get more information about the equipment which we have. So we have Dr. Premnath with us. I request you to kindly come here. And so we had very wonderful talks by Dr. Taruna Arora. She had sent a pre-recorded video, and then was Dr. Jyoti Iyer, and followed by Dr. Sami Sangeethra. So I would request you to first felicitate our chair, Dr. Shirmuli, and then our speakers.
Yeah. First of all, my apologies for not being here. Today turned out to be a very busy day. We had a whole series of pitches happening. There were people from different parts of the country, investors also looking at some of these things, and I had to be there. So sorry about it, but I thought I'll join in the discussions, panel discussions. So first of all, thank you very much to all of you for participating and also contributing. This actually, I just wanted to add a couple of words to what Shrikant also said. One of the things to keep in mind is, as Venture Center, we have also been continuously evaluating our role, right? We do, of course, support startups. That's what we got started with. But there's a larger activity, which is creating an innovation ecosystem. And there, it's not about the kind of entities. It is about creating an ecosystem where everybody can play. If you're not able to do that, you can't really progress, in my opinion. So that's one of the things which we have always kept in mind. So our effort is to try to create those ecosystem components one by one. We started with simple facilities, more specialized facilities, mentoring, financing, IP, tech transfer, regulations. All of those are trying to plug the gaps. So we are quite positive and very optimistic about biopharma. Okay, we think India and more specifically Pune has a big role to play. Okay, and we also think that it's going to be driven very differently than perhaps some other domains in India. There's also a large opportunity emerging in similar to, say, what generic pharma happened many years ago, but we may short circuit the process and also go into certain other things which we may not have expected will happen. So I'm hopeful about that. We do understand the challenges, but there are also opportunities, and right now might be the time to sort of play the game, right? So our job is to create mechanisms or create the ecosystem which creates options. Options are, if there is a situation where you need a solution, you should be ready with it, right? That doesn't happen unless you have created a base to work upon. So that's what we're trying to do. So biopharma analysis, Center for Biopharma Analysis is an effort in that direction. In some other domains also, sometimes we have to bet on certain domains. So we are betting on biopharma. We're grateful for Shrikant to join our team and this effort to make it possible, and all of you in supporting us as well. We are very keen on it, and we will build on it. We look forward to your advice, expertise, and the inputs in shaping it further, and also networks. So all of you, I hope, will be continuously contributing to this as well. So sorry, I took a little longer there.
You can take out your mask.
I think I have to do it for the photos.
Yes. So you have Sarah? Okay.
So we have our. Thank you so much.
Thank you so much.
I didn't want to run a session on your book.
Thank you.
Please.
I requested him to get few copies. Dr. Jyoti.
Dr. Jyoti, thank you very much.
And Dr. Sami.
Thank you so much. Great to meet you. Okay.
Thank you very much. We'll now begin with our panel discussion. So I request my team members to kindly help us with the arrangements here. And we'll stop sharing the screen now.
You're using that actually.
This one.
So we don't want to take a 2-minute break or 5-minute break?
You're running late.
We're running late. Okay. So what I thought we would do is I don't know how many of you know what I like. I don't like to do anything normal. So I like to do something abnormal. So what we're going to do, and the students don't ask, as you guys ask great questions. So these students ask great questions, right? Let's give them a hand. Like, this is amazing question. And they're wearing pink shirts. More power to them. So what I'd like to do, maybe we can do the first traditional way.
Yeah, but before we do that, maybe Yashodhan, can we have a group photo of all the people who are joined online? Can we request them to kindly put on their videos? And while the speakers are here, we can in background have a group photo with them. So you all can please be seated.
Sure.
Yeah.
Yeah.
Yeah, he'll do it.
I think Jyoti will come here.
Yeah, by the time people switch on their cameras. Dr. Srikanth, please. Yashodhan, you can tell us, when we'll wait for 2 minutes for Dr. Iyer to join.
We request all participants to kindly switch on their cameras.
Photo?
Yeah.
Okay.
Are they all seen now?
You have a maybe expand it a little.
If I move it over here, let me just give it a try.
Go back. It's okay, just expand the picture.
Yeah, Dr. Iyer, please join us. So are we good to go? Group photo?
Yeah, I think
Yes.
Yeah, sure.
it's already happening.
There's something online that is some magic that's happening online. Great.
Thank
Continue.
you.
Yeah.
Yeah, let's continue.
So do we just sit here and do the panel discussion?
Yes, yes.
Okay. Is everybody visible and all
Yes,
that?
all of
Okay, great. So we hadn't really planned on what the panel discussion was going to be. So I love when you don't plan. I can do whatever I want as the chair, right? So I was thinking a couple of things. First, let's open it up for just general questions for about, how much time do we have for this panel discussion?
20 minutes.
20 minutes? Okay, so unless there are like burning questions, let's do first 5 minutes questions because lots of questions have already been answered. And so the next 15 minutes, we're going to do something unusual in which you are all going to be on the firing line. Okay. All right. So please let's any points to discuss, any points, just open-ended questions on all the talks. Yeah, yeah. Mike, please take the microphone. And please introduce yourself as you ask. We'll keep this one here for the audience.
Do you want to use the color mic?
Yeah, I can.
Hello, so good evening to all. My name is Evan Ali. I am a PhD student with Bharati Vidyapith, which is in Pune. So actually, my question was that this it might be slightly off topic, but how do you navigate patents as an academic when you want to take your products to a global market or something like that? Because what I'm working on right now, there might be a patent in the future, so something like that. So just if you have so many.
Maybe you can specify a little bit. I can take that.
Yeah. Why don't you take it on?
It will be in the vaccine department. So my PhD topic is about.
Elaborate a little bit more. Spell it out a little.
Okay, so
it's a very large topic.
Okay. So we're developing a vaccine candidate for a virus called respiratory syncytial virus, and we're hopeful that as everyone, we're hopeful that it might be successful, and we want to find a patent for it as well. And just wanted to ask you about how do we navigate it, with the stages of patent filing, and if there are any documentation required for that.
Okay, so I'll take this on as a sort of guest in that sense, but. So, by the way, I used to head IP for NCL, and we are into technology commercialization. So this is our bread and butter anyway. So happy to talk through that. So first of all, when you're looking at specific ideas and new technologies, the first task to do, whether you patent it or not yourself, the first task to do is to ensure that you have freedom to operate, right? Not necessarily a legal opinion, but at least knowing that other people have not sitting on patents which prevent you from progressing ahead, right? That's what it means. So basically you figure out other people's filings which are going to prevent you from progressing ahead. You don't want to find out after 3 years of hard work or 5 years of your PhD that somebody else owns it, right? So that's the first task that one typically does. That means you try to see that you have some space. The second aspect is you yourself patenting it, right? Now, if there is an invention there, or any, now, by the way, simple thing to remember for many students is, see, most inventions are incremental. Okay, don't expect that it will be path-breaking, dramatic, and so on and so forth. That happens once in a lifetime. Okay, very rarely. Most inventions, you have to basically spot what is an inventive idea out there, right? So typically inventions consist of two parts. One is a problem and a solution, right? You define a problem and then you propose a solution for it. If the solution is novel, okay, you have something which could be patentable. There are a few more things which I won't go into, which are related to the patent law. In every country, the patent law changes, right? So you have to know what is patentable out there, right? Typically, a simple way of thinking about it is you have something new, but it cannot be something which somebody can easily extrapolate. There's something surprising about the development that you came about, right? In the context of what other people know. Okay, so that's the simplest version that I can tell. So you should be looking at patents as a way to create a competitive edge for a person who's going to invest in your own technology. So if you're a student, why do you file a patent? One of the main reasons is you know that your field is, whatever you're developing is going to need a considerable amount of investment to take it ahead, right? Now, it's more attractive for somebody to invest if you have an edge, right? So that's one of the reasons. So I tell people that for most academics and researchers, the biggest reason to patent is to attract more resources to your own idea. Okay? It's not about getting a label and putting it on your resume. Okay? So this people miss on. Now, once you have created something which has created an edge, the person who is looking at it is going to see, if I'm going to invest 10 years to take it to market and invest, say, 100 crores, right? After 10 years, can I defend it, right? And in pharma, you know how it works, right? Typically 10 to 12 years, you're developing a product. You have the last 8 years to make money. And after the 8 years end, there's a cliff and the price drops in no time, right? So you have those 8 years to make money. Now you have to protect that 8 years as much as possible. And that comes to your patent protection, right? So typical process is, I need to brief, I realize. So a typical process is you create a portfolio of IP that you can protect. There could be small things which you protect. There's a strategy behind it. It's a little deeper discussion, but you can protect those and take it and create a portfolio which is attractive to a licensee or a buyer of the technology so that they invest money in it. Okay. Now that's about broadly the picture. Okay. It's no big deal. We have a patent facilitation here, an IP facilitation even here. We also run programs where you can learn about this. You can come and talk to our patent agents. It doesn't cost any money. Talk to them first, understand what can be patented, what can be protected, and then take it ahead. Okay. It's another one last thing I want to tell you is people think that it's only a big boys game. Okay, it is not, right? You can actually file patents, find those niches where you can file. Okay. You should be able to find a niche and can be valuable as well. You can also, the real money goes out at the later stages when you're maintaining a patent. But by then, if you have found a licensee or an investor, they will support you. right? So you should just focus on just taking care of filing first and filing before you publish, right? As a student, I think that's what you need to ensure you do, right? And there are some other simple practices. Can I take one more?
Take your time. This is very valuable for the students.
Okay. If some of you want to read a little bit, a good case to read is the CRISPR fight that's going on right now. Okay, so you know that there's one group on one side of University of California, Berkeley, right, and University of Vienna, and one other party which is involved in one side of the filings. Right? That's Doudna, the Nobel laureate, and Charpentier's patent, right? And the other side, there's Broad Institute from MIT and Harvard, right? Zhang, who has filed those. Recently, there's been a news which basically there's been a court ruling which basically says that it's for the one that matter, which is for the eukaryotic cells, right? The patent actually, the one that stands up is of Broad and MIT and not of Berkeley. That's a big setback for Berkeley, right? Because they originated the tool, that's CRISPR, but they did not protect adequately, or they did not demonstrate the results for eukaryotic cells, which is mammalian cells, which is what all of you will be using for many applications. Okay, so the lesson from there is couple of them. One, if you go and look at an article, which I can send you if you're interested, Smita, I can give a link. The lab notebook was very important. Okay, of the postdoc who did the work, right? There's a way you write the lab notebook, the way you record it.
Not has been discussed about it.
Okay, that decided the fate of this particular protein. There was something called an interference proceeding where they had to figure out who was the first person to invent. Okay, and invention, there's a formal definition to it. It's a legal definition. It's not a casual definition. Okay, that failed, right? It's not the student's fault. Postdoc did a good job, but the recording clearly showed that they had not firmed up, they had not reduced to practice the use of CRISPR for eukaryotic cells. Okay, whereas just a month later, right, Broad Institute had done it and filed the patents. Okay, so it's of course going to be a big battle, but behind it, behind the scenes, they have licenses. There are several startups, including some you are hearing in the news in biopharma, Intelia, for example, right, which is getting into. You must have seen the genetic diseases that they're curing with gene editing techniques. Okay, those kind of examples. Now they are going to be quite. There are people who are going to suffer if they don't have a license. They'll have to go to Broad Institute to get a license. Okay, so this dynamics always happens. Now, in all of this, you must be noticing that these are none of them are big companies, right? So universities, right? So why not you? Why not Bharati Vidyapeeth? Why not you, right? So I think it's possible, right? It's about ideas. It's about filing those in time, spotting them early. Very often people even miss that opportunity. They don't think it's important, right? One of the biggest challenges in innovation is that people don't realize that a topic is important. They don't realize that you can't pick winners actually. Okay.
Quick, another reference is a book, highly recommended. It's written by Joanna Broger. She is a professor at Cornell University, and the name of the book is called $1 Billion Patent. Highly recommended. It's very easy to read, and in addition to the resources, that book is very good.
So sorry, did I answer your question?
Yes. So it was lovely to hear all of that.
Please do file patents, irrespective of what other people say.
More questions,
So this is in general actually.
I request you to kindly introduce yourself.
My name is Uday. I work as an independent consultant for the BioPharma.
Okay.
I have 25 years of experience, worked with the various organizations. In fact, I introduced Corning in India, Thermo Fisher, then the last one was the Life Sciences, and last 12 years I am independent. Basically, I don't like work same what you said. I'm really abnormal as far as the technology is concerned, and my appetite is always bringing the new technology, novel technologies to the area, and I'm successful with 3-4 acquisitions also, which I introduced, and then same company fall and Thermo Fisher acquired those small companies. Okay. So that is my. Yeah. So my question is that basically looking into the government, because we have seen the COVID phase and it was really horrible and nobody can expect that this will happen. Whole world was wrong. So thing is that the government spending a lot of money on the research. ICAR, ICMR, even though the CSIR, all these are the the authorities who have all the rights and the government. But I feel specifically about bad about one thing. That as we know we have the best experts. Means if we are talking about the NIV and all the great virologists, they know the virus better than any other in India. They do a lot of research, but it never comes as a process development or till that level. So that if this sort of the pandemic situation or any situation like this, they can utilize and do the tech transfer to the the companies and utilize whatever the resource we have, and we pay for it. Means the government or taxpayers' money. That is first thing. Second thing, I feel very bad when the one company you might be knowing about the Hindustan Latex. It was previously a condom company. Government given it's a subsidy of government. Government given them the 300 crores. They make beautiful plan, which is for the rabies vaccine. Even though I don't understand when the NIV they have done a lot of work on the COVID or vaccine sort of thing. They have the everything about that. It can be transferred to the government facility and government make their own vaccine manufacture. It may be, I'm sorry, that may be the hot question, but I don't know.
I will let you and Smita decide whether it's within the scope of discussion, but I'm happy to chat with you separately.
Yeah, because
I can do that.
Yeah, no, I think time is running out, but maybe it's a good conversation over beer.
Okay, a short answer. During COVID, okay, many of those facilities were actually put to use.
Yeah.
Okay, you may not hear about it, but they have been put to use. I'll explain some of that to you. I'll give you examples that some of them have come useful. That's a different matter. Maybe we should have done 10x, right? But the 1x happened. Okay, so I can give you some examples of that.
Yeah, but thank you for bringing that point up. So, any last few questions? I can just ask questions.
So so I'm Bhagyashree Mane. I'm from CSIR Pune. I would like to add the point. As sir said upon that IP, so I have attended all the lecture series and we have benefited because we are NCS students. But yeah, other students also enroll. So you can get the link and other students also join. So that's the add-on. And my question is to ma'am. Ma'am, you said like high risk to the ligand and the low risk to antibodies. So can you explain that? Why in the drug development? So why it is? Okay. Means the ligand compounds, they are high risk to drug transfer, means the transfer the drug for the toxicity stage. And for antibodies, it is the low. So why it is?
Maybe it's an endogenous protein.
Are more specific compared to the small molecules?
That's
actually that is the answer.
That is the answer.
Yes. I think your question is about the toxicity of small molecules.
Yeah.
And the safety. I mean, as compared to that, it's considered that there are less side effects for vaccines. Right? That's it.
Yeah.
The main reason is the specificity of the antibodies. I mean, they're actually similar to what our body needs. They are not new. Whereas you look at chemicals, they're completely new and foreign to the body, right? So the body has to process it. I mean, these chemical entities are very, very different. That mainly brings about toxicity.
I think of it residual level.
Yeah. Can I interrupt because we have only a few minutes left? I want to do this small exercise for the students. So if we can take it over beer.
I didn't know.
Not in NCL category.
Okay, I'll just give it. Thank you, Bhagyashree, and the excellent questions you asked during the course. Thank you. Any burning questions from anyone? Please.
My name is Darshan Mahali from Pune College of Pharmacy. As we know that analytical method development is a key stage in any product development. Recently, USP released the guidance for analytical quality-wide designs. So is it applicable to novel biological products or biosimilar?
I think any analytical development guideline that you see is applicable for all the antibodies, be it for biologics, vaccine, novel biologics, biosimilars, pharma drugs. It will be applicable.
Is it mandatory for the?
So the US has given a guidance. So when you go through the guidance, it will be it will give you a kind of a backbone of how you develop a method. Okay, and how you validate, etc. afterwards. And that's, they have noticed that R&D is not following harmonized when that's why they do that. So it is, I think, good to follow.
Actually, if you're talking about the United States pharmacopoeia, it is not a statutory body. It's actually a commercial money-making business for 200 years. Right? I mean, I'm not saying in a negative way. I'm just saying that it is not statutory. So if there's a USP guidance, there is no requirement that you have to follow it. As Jyoti mentioned, you have to demonstrate to the FDA that your assay that you used was fit for purpose for the application that it was required. Okay. But anyway, those guidances are very useful. In college, you should study everything. Yeah. Some syllabus there. At the back, I think there was a question.
Myself, Ravindra Wavade from Deegar Patel College. My question is, if you conjugate a drug with antibodies, will it come under the biologics?
Yes.
Sure.
Yeah, so one of the product is Kadcyla, where the trastuzumab is conjugated to ADC, that is Emtansine, that is the drug, Cyclosporine. So that is coming under the biologics. Because the primary action is driven by the monoclonal antibody, and then the drug is released for the cytotoxicity.
If the case is, if we want to improve the specificity of drug by conjugating with the antibodies, so in that case, the drug is the main ingredient, and antibodies only for to increasing the specificity.
So if you in India, again we'll have to discuss, but for example, if you go to US, there is a combination department. So you have to apply for designation, and when you apply to them for designation, they will give you whether your active part is your drug or your molecule or both. If it is both, then they will give you a designation that this will fall under biologics. However, it also will, the sections will also go for the CEDAR or CBER to review. So you have to file for designation in that case for this.
I see Taruna on the line.
Yeah.
Taruna, can you hear us?
Yeah, I can hear you.
Is it possible to
I can hear you. No, I could not chime in and for some of the earlier questions. I was not muted. Yeah.
Yeah, no, your video played really well. Thank you. That was very, very good.
Yeah.
And it's 7 o'clock here, and so we are coming to the close. But I think since you've come and we're all ready for something.
There was one question.
There was one question for her. There was one question for you, Taruna.
Okay.
Smita is looking for it.
So there is a question: is it compulsory? Is it compulsory to conduct in vivo toxicity or safety studies for biosimilars?
Naren, can you repeat the question?
Okay. Is it compulsory to conduct in vivo toxicity or safety studies for biosimilars?
Oh, okay. Well, so far as we know the guidance, no, it is not required to conduct preclinical safety or toxicity studies as long as your biosimilar molecule has been characterized and shown to meet the criteria for product attributes that should be similar between innovator and the biosimilar. So if you have really done very well, thorough studies for product quality attributes as well as for your in vitro functional characteristics, you can show that you are managing like glycosylation or some other modifications, and they're not really going to have any impact on activity or safety by in vitro assays, then you can skip. No, agency doesn't require. When they initially started, when the first product came for, came for application of biosimilar, I think for everyone, including agency, it was new experience. But as now they have become more experienced and mature, and then, these studies are not required.
All right. Thank you, Taruna.
There's one more.
Okay.
Yeah. There's one more question for you. What differential pre-IND considerations are for oral biologics?
Oh, I am not sure. That I don't know if someone else can answer.
I think if you look at from the pre-IND perspective, anything is like there's no requirement. There's a specific questions, the specific enabling studies that you have to do, and go for agency to ask that questions. Basically, if you look at from oral perspective, the burden of toxicity, etc., will be little less. So you may have to do less tox studies, and the clinical will be easy rather than going for a subcut or IV studies. So the parental studies will be in the pre-IND difficult to convince, but if you have oral biologics, then I think it will be very easy.
Yeah. And any other question?
So there is a question like, what are the knowledge skill attributes which I need to learn? So there's a question by Dr. Santosh, who's resident in clinical pharmacology from JIT MER Pondicherry. So which skill attributes which I need to learn in order to collaborate better with biopharma scientists to enhance efficiency in the drug development process?
You want to take collaboration with that?
Yeah. That I can, but I thought he needs to. It's what skills, right?
Skills,
yeah. What skills do you need?
Maybe technical skills from in academia. I think the question is more about he's in academia.
Yeah.
If he wants to talk the same language with industry, right? What are the skills that he needs to learn, or what language he needs to learn, right? The wavelength has to match.
Yes.
Yeah, so now you have been in the industry.
I'm the moderator, so I can tell people to answer questions. Yeah,
Would you like to comment?
So I think we talked about GDP and
yeah.
So, one of the please.
No, yeah. So I mean, it's a simple answer. I think there's no magic in industry. I say industry में कोई we are not aliens over there, completely different, and we don't speak a different language. हम लोग हिंदी इंग्लिश ही बोलते हैं. सब कुछ same है, right? But I think if there are any things that I'll say a few things. Maybe Jyoti and Samir, you can add a few points. I think there's something about rigor and accountability. अगर किया नहीं तो पड़ेगा. मतलब पड़ेगा ना. There's a lot of accountability. And one other thing that is it's much more regimented, meaning I have quarterly goals, monthly goals. I have a boss. I have to report to my boss every X amount of time. There's a lot of rigidity, right? So I'll make that. Jyoti, you want to add anything to that?
I think some of the notions that students have when they come to the industry is that you get to do science from the starting to the end, that is from the cell line development until the drug product. That is not true. I mean, you need to be ready to work only in a very small area for a long time. Like it could be you're doing the same method, or you're developing analytical method for like 4 to 5 years. So the skill that is required is to be patient, to be able to find the role that you are contributing as a scientist in the larger picture.
Yeah.
So be able to motivate yourself and.
So I think industry is as you rightly mentioned, it works on projects. There's nothing academic or research going on there. Each project, every project will have a deadline. So I think interacting with industry people more will help the students and researchers, because they understand then the language that, okay, once we say that this cell bank has to be prepared and characterized, and then this batch and then bioreactor and all those things. So project management skills, those are very much available on this. So that is what I would say.
Right. I mean, Srikanth, this is your basically exactly what you said. I think it's more regimented and everything is a closed goal, not open-ended goal. So this is major difference. We will have a weekly goal or monthly goal, yearly goals. They need to be met, no matter what. If that's failure is acceptable, if it's done correct. So I think his question is at a different level. So he wants to collaborate with industry scientists and able to talk. Yes.
I can.
I have not said that. Collaborate.
Okay. So from what I have experienced, the first thing is I think industry is not a monolith. Okay. Not all companies are alike, right? So you have to understand who you're talking to, and also within a company, which role are you talking to, right? So on one side in biopharma, there will be, say, a company which is spending 40% of its expenses on R&D, but that's mostly in the US. Perhaps like Vertex, for example, I saw 40%, right? That's unbelievable. On the other hand, there might be somebody else who is focused more on manufacturing, more on line functions, and R&D might be a relatively smaller activity. Also in R&D, there'll be different types of R&D, right? Some which are creating new businesses, some which are focused on improving current businesses, improving the profitability of current businesses, and so on. So there'll be more process R&D and so on. So first, I think it's important to understand where you're going, right? So now, all said and done, there are companies which are looking at R&D as a core activity, like some of the companies which are betting on new technologies. For them, they have a very different way of looking at how they build technology. So they will want to build new ideas, create new IP. They will look at what the investors want. It's more investor-driven very often. Okay, so somebody like Moderna today might be looking at what pipelines they have, and how many pipelines they have, how far it's progressing. Stockholders are looking at it differently. If you look at somebody else, maybe Serum Institute or somebody else of that kind, it might be very, very different in terms of the kind of molecules they are focusing on, or vaccines they're trying to focus on and take it ahead as well. So as a person in academia, you might want to learn, first of all, I think in some cases you need to explain to people why. One of the gaps I see is people don't know how to explain why their work is important. Okay, simple things like that. Can you explain why you should be doing it? What is it in terms of value creation for people? So the business managers typically want to hear that, right? And that's important learning from that. When you're dealing with technology folks, very often in many companies, it will be, as I said, projects, right? So you need to be able to pitch internally a project, get it going, manage it with different resources. Project management, of course, comes in, but it is also about knowing some amount of technology strategy. How do you know what to pitch? One problem I see amongst many academics, which Professor Langer at MIT says, is that, look, all your life, we give you problems and you solve them, right? You never learn to create your own problems, right? But in life, that's what matters, right? That means you have to define problems, right? So that is another task, in my opinion, which you need to learn, right? And that is valued very often in certain roles in the industry. Of course, there will be functions where you have to just be the expert, and in which case you have to have patience. Somebody who doesn't take time to build that expertise over a period of time is not going to be valued. But so there's a role for specialists, there's a role for people people who are in the interface and somebody who's a generalist. All of them are possible.
Sorry, just want to extend that point. I think this question is there any bridging between the industry and academia today? Is there a place where students can go understand more about it? So I think that's the question.
Yeah. So we do some amount of training in such aspects. For example, there's one of our mentors who has previously been with McKinsey and worked with Abbott, who actually came and taught about how industry evaluates projects and taught that as a course to students. Hopefully we'll run it again soon. Okay. So, but it requires you to think not only about technology, but also about a little bit of financials, understand how it creates value, right? So that is one kind of thing. The other is also in several companies, people don't realize that the patent literature is actually a very large body of literature. It's in fact larger than the scientific scholarly literature, and people have never seen it. So if you're preparing for the industry, it might be a good idea to at least read one or two patents, understand how it looks, what is the useful part of a patent, what can you get out of it, and so on and so forth, right? So that's a good learning if you want to learn that, because I think you'll get a lot of information from that literature as well. And so these are some bridging kind of, getting to speak the same language kind of. One of our colleagues, one of our mentors here called Ravi Sarangpani, he runs a specific program on just project management, okay, where he teaches people how to do detailed project management with all interdependencies and so on and so forth. If one goes late, what all does it influence later, right? So entrepreneurs do that actually because it makes a big difference to their ability to raise money. And regulations, can you understand those? Can you tell me, Indian pharma companies, what is the biggest determinant of share price? Like you hear in the news, right? Somebody's share price dropped. Why? Probably the US FDA came and said that this plant cannot supply to the US, right? So clearly, the value depends on regulations. So you can't discount that. If you don't know any, as an academic, you can't say that I don't know enough about regulations. You have to know about it, right? That's going to be a very important determinant, right? Clinical trials results are tracked so heavily by the venture capitalists in the US, right? I follow a couple of them in the Boston area, okay? And they track every disease, which are trials happening, which one failed at what stage. You can see it on Twitter, by the way, if some of you want to follow, please do, right? So you know what's happening. So if you are an academic and you want to be in, say, cancer therapy or immunotherapy and look at all cell therapy, gene therapy, whatever it is, you need to know what's happening. So that's different from what you learn in a textbook, right? That's a foundation. After that, you have to figure it out yourself. Right? So these are some things that you might want to consider. I don't know the answer.
I think we'll have to wrap up now.
I just have one small question. This is great that people are asking so many questions. So I'm Satish Pathak, running a startup in the field of industrial automation. But having some context from my friend, somebody I know. Now we all talked about this biopharma and the pharma industries and all that. Would you like to have a symposium for medicines under Ayush or the essential oil-based kind of medicines, which also are equally effective and less threatening comparatively? So would you like to have any symposium on that?
Question is to me, I think, right?
Yeah.
Yeah. No, definitely. All we are mentoring already startups in all kinds of medicine as well. But everything has to be technically sound.
Sure.
Okay. So that there's no excuse there, right? So one of the problems we have is that, by the way, I really think there are great solutions available in traditional medicine. but you have to be credible in what you do, right? Which means that whatever, one of the problems, there are two problems right now which we are trying to see what to do. One is raw materials quality and standardization in traditional medicine. And the second is clinical trials, right? Now in India, generally speaking, clinical trials area is weak, right? So there's a bigger activity needed to strengthen how we do our clinical trials. Right? Unless we could do credible clinical trials in India, right, people will not believe us. We can show some small things and get away with it, but that's not good enough. You know what's happened during COVID also. Yeah. So many guys did so many trials, so many medicines were tried. By the way, it's in hundreds. We have done a catalog of it. You're aware of it, right? How many produced one credible candidate, drug candidate, right? So I think we need to learn to do it systematically, right? And that requires some effort. So we are trying to see how to build those as lasting capabilities.
I asked this because
we'd be interested in doing the symposium. That's short answer.
Okay. I think in the interest of time now, we can wrap up.
Yes.
But I have one thing. No, I was going to do a much larger thing, but I'll do a small thing with the students before the class. Before the session, I'll ask the students to ask questions. So maybe we'll ask you. Tell us 3 things that you learned today that you did not know before.
Mics. Circulate.
And then we'll close. Okay, Smita?
Yeah.
Okay, no, tell us 3 at least.
Very short.
And not you, maybe not you. Maybe
She asked a lot of questions.
जिसने question नहीं पूछा. जिसने question नहीं पूछा. जिसने question नहीं पूछा.
उससे बिल्कुल बात नहीं की है.
Test. Didn't know there was a test. What did you learn? कुछ भी बोल दो यार. हमको क्या पता तुमको पता था कि नहीं?
Hello, sir. I am a M.Com second year student from Dr. Devar Institute.
Nice.
As I am a QA student, I know the importance of documentation. But from this session, I know how much data integrity. And one statement is that data integrity is regarding the mindset. Data integrity or the documentation is related to mindset. So,
and did you know about the word ALCOA?
Yes, no, sir.
There you go. You learned ALCOA.
Yes.
Okay. What? Okay. Don't forget ALCOA.
Yes.
Second thing. 3 things we want. Anything else you learned that you did not know before? Either आगे, oh my God, ये सीख लिया. Yeah, Smriti.
There is high advice fees charged by EMEA.
Good. Last one. सब कुछ पता था पहले से. Yes.
30-35 comparison tests. I was not aware of this.
I have one point to add.
Yes. Is there
I gave that mic away. So.
Yeah.
Okay. And now
I think this is a very important thing what Dr. Narine asked today. How many things you have learned today? If you can invest 200 rupees on a diary where every date is written, and if you write 2 things what you have learned today, you will be. I have asked my daughter to do it, so you can also try to do it. Only 2 things, not 3. Just 2 things.
That's a great solution. Thank you very much to all our panelists. Thank you, Dr. Premnath, for joining in. So we are wrapping up now, and thank you, Dr. Taruna. Your presentation was very great, and in a very lucid manner, you explained the whole development pathway there. And thanks for joining at this time, early morning there. Yeah, thank you very much. And thanks to all the participants who are online. Yeah, please go ahead.
No, thank you. Thank you for this, opportunity to be part of the panel. Yeah, it was good.
Thank you. And thanks to all the participants who have joined us online, and thanks to all the participants who are here. Please join us for the dinner. It's being served at the cafeteria. So our team members will escort you there and guide you. And thank you again to everyone. Yeah. Meghna, we are good to close the Zoom meeting.
Okay.
Thank you so much. Because of that only, I get doing
That's